Good morning, ladies and gentlemen, fellow speakers, friends of science, technology, and innovation, and good morning to our audience joining us online from all over the world. I am today's first host, Qin Lang, a postdoc from Peking University. It is my greatest honor to welcome you all to the webinar on 2022 Beijing Brain Conference for the Postdoctoral Forum for Advanced Interdisciplinary Studies. This event is held by Academy for Advanced Interdisciplinary Studies and Peking University, Chinese Institute for Brain Research in Beijing, Center for Life Sciences, and Microbiota.
Could you hear me? I cannot hear you.
I can hear Min, but cannot hear the host.
Okay. Should I suppose to start talking?
Okay, can you hear me now?
Oh, yes.
Yeah.
Okay, there is something wrong, obviously. Okay, I will restart, okay?
Okay, go ahead.
Thank you. Okay, so sorry for this little accident. Good morning, ladies and gentlemen, and fellow speakers, friends of science, technology, and innovation. And good morning to our audience joining us online from all over the world. I am today's first host, Qin Lang, a postdoc from Peking University. And it is my greatest honor to welcome you all to the webinar on 2022 Beijing Brain Conference for the Postdoctoral Forum for Advanced Interdisciplinary Studies. And this event is held by Academy for Advanced Interdisciplinary Studies at Peking University, Chinese Institute for Brain Research in Beijing, Center for Life Sciences, and McGurven Institute at Peking University. And this is also part of a series of online events of Zhongguancun Forum. Well, as you all know, postdoctoral time is really a critical period in a scientist's career path. And this very forum, particularly, is aimed to provide a platform for young researchers from fields of multidisciplinary studies to communicate, inspire, and spark ideas. And before everything, I'm more than pleased to introduce our advisory board members today. Okay. So Dr. Chen Jian from Chinese Institute for Brain Research in Beijing, and Dr. Guo Qiang from School of Life Sciences at Peking University, Dr. He Aibing from College of Future Technology at Peking University, Dr. Luo Mingming, co-director of Chinese Institute of Brain Research in Beijing, Dr. Li Yulong from School of Life Sciences at Peking University, Dr. Song Yan from School of Life Sciences at Peking University, Dr. Song Chen from Academy for Advanced Interdisciplinary Studies at Peking University, Dr. Wang Chu, Vice Dean of Academy for Advanced Interdisciplinary Studies and Professor of College of Chemistry and Molecular Engineering at Peking University, Dr. Wang Wei from School of Life Sciences at Peking University, Dr. Wang Zheng from School of Psychological and Cognitive Sciences at Peking University, Dr. Zhou Peng from College of Chemistry and Molecular Engineering at Peking University, Dr. Zhang Yan from School of Life Sciences at Peking University, and Dr. Zhou Yang from School of Psychological and Cognitive Sciences at Peking University. Thank you all for joining us. And first of all, let's welcome Dr. Luo Mingming from Chinese Institute of Brain Research in Beijing and Dr. Wang Chu from Peking University to give us the opening remarks. Welcome.
Okay, great. Thank you for this opportunity. It reminds me of the age. Once I get to a point of certain age, I will be invited to talk about something. Let's use this before the really important talks. I'll start with a joke, maybe a real story. When I was, I heard when I was a graduate student at UPenn in Philadelphia. I remember it's like, once a postdoc walked in the street at night, somebody came up and pulled out a gun and pointed the gun to him and say, give me all your money or your life. The postdoc answer, just let me go. I don't have either money or a life. It's just like this, the common feeling for postdocs. It's you feel it's tough, but it's really looking back, it's the golden years for all researchers, it's kind of like point of no return. Right? Once we have seen like many national leaders with a PhD in science or engineering, but you don't. At least I have never heard of a national leader who has done a postdoc. That means when you do a postdoc, you will continue to do research. It's really great, when you're doing a postdoc, you don't need to worry about grants. You spend all your time doing experiments, and that's the best part of it. You still do experiments, and your ideas make sense. Once you have your own lab for many years, just like many of us here, we read papers, we look at status, we come up all these fancy ideas that's maybe fail 99% of time. But when you are still a postdoc, you know your field. You're the best experts in the entire world in your topic. So although it may seem hard, in terms of the competition and the money, you can do a lot of stuff. You are still very energetic. And I want to congratulate all our postdocs speakers today. We have 18. The fact that you have been chosen to speak here means that you have made some really good achievements. And this is also a golden year, the best time for our postdoc in that life sciences, at least like neuroscience, brain research, is expanding dramatically. Everywhere we are hiring, like Cyber, the Chinese Institute for Brain Research in Beijing is hiring. And I'm very confident that Peking University and Tsinghua University in Beijing are also expanding. And in cyber, we already have 32 labs and 10 core facilities. We remain the strong believers of exploratory research. We provide very strong support, both in terms of funding and the technical support. We have 10 core facilities. And we'll just allow our scientists to focus on really important questions. So, when you are looking for a job, remember, explore cyber and Peking University and Tsinghua University, or other top research institutes in Beijing. As a matter of fact, I think some of our PIs in cyber are in this room, and we are very motivated, and we are very excited to hear your talks and see whether we can find good matches. Okay, I'll stop here. Welcome again.
So I guess it's my turn. Oh, your microphone is muted. Okay, I'll take over. Good morning, everyone. I'm Chu Wang, and I'm currently serving as Vice Dean of the Academy for Advanced Interdisciplinary Studies, short for AAAS, at Peking University. So the organizing committee asked me to also give opening remarks, and also in English. This is kind of a very special experience for me. But first, I would like to send my sincere greetings to this wonderful event on behalf of the AAS at the Peking University. Since this is also part of a larger conference, the Beijing Brain Conference, as a sub-symposium, so I just want to give maybe a brief introduction on what the AAS is and the history about this institute. So our academy was founded on April 4th, 2006. So that's almost 16 years ago, more than 16 years ago. And at Peking University, it was established as a pioneering institute at Peking University with the goal of expanding our knowledge of the fundamental science through the cooperation of multiple fields. In the past few years, AAS has been devoting in the construction of new disciplines, development of frontier researches, and education of talented young minds with integrated perspectives. So in AAS, we believe that advance of modern science and technology urges the interdisciplinary interactions as a major platform for the interdisciplinary research and education at the PKU. AAS is supported by extraordinary academic resources from multiple departments and college of the university. So currently, under our academy, there are more than 10 research institutions, including the BioMedX Research Center for Life Sciences, Center for Quantitative Biology, Center for Nanoscale Science and Technology, Center for Fundamental Imaging, Center for Environment and Health Center, and the Center for Health and the Philosophy of Science, and also the Beijing Brain Institute, Beijing Brain Institute, CIBR, Chinese Institute for Brain Science at Beijing. So we also have affiliated a graduate program with different institutions. And the research institute covers areas of mathematics, physics, chemistry, biology, and medical science and engineering. So this is truly a field for the interdisciplinary studies at Peking universities. And all this work has been possible due to the great effort from a group of brilliant professors, postdocs, and graduate students. And also, they generate many award-winning scientific breakthroughs and technology innovations. So in the future, AAS will keep on playing an essential role in the development of forefront science and help PKU with its aim of becoming a world-class universities. As Professor Luo just said, right, today is a postdoc forum for the interdisciplinary studies. And also, in my personal opinion, I believe that postdoc is a major workforce in performing the interdisciplinary researches because you already, become a master in one field, and then this is the best time for you to explore across the border to explore another exciting field and then generate new ideas and generate new hybrid. So I think postdoc is also the best stage in one's career to do the interdisciplinary studies. My personal experience, I benefit from doing the interdisciplinary studies by extensive trainings in computational structure biology in my PhD training. And when I decide to do a postdoc, I want to try chemical biology. And then that experience made my independent career more uniquely distinguished and setting me up with a more open mind to explore science. So at AAS, we not only have the interdisciplinary graduate programs, we also dedicate support and training for the postdocs. Just to mention that we have a very strong and prestigious fellowship to support the interdisciplinary postdoc trainings, and we will continue to do so in the future. If I remember correctly, this is the third time we start organizing, since we first started organizing such a symposium for the postdoc researchers to communicate and exchange ideas. And also this year, this symposium is honored to be part of the 2022 Beijing Brains conferences. So in the end, I just want to thank the organizers, the staff members, and the advisory board, and all the organizing committees for their collective efforts for making this event possible. And I also want to thank the Chinese Brain Institute Beijing for their generous support and the co-sponsorship for this wonderful forum, and thank all the audience online for participating. And I wish you all enjoy the rest of the day for this great science festival. Thank you.
Thank you. So, we are really grateful for your kindest remarks, and thank you, Dr. Luo and Dr. Wang again. And actually, all of the postdocs and our colleagues, we're really having a great time here at Cyber, at AAS, and Tsinghua University. And I believe all of us will enjoy today as well. So now we shall begin the first session of our forum. So our next speaker is Dr. Wu Ming from Harvard University, and she will be introducing her work on the genetic mechanism of selfishness and altruism in parent-offspring co-adaptation. So welcome, Dr. Wu.
Thanks for having me here. Parental care is an altruistic behavior. Altruistic parents provide their offspring with food and protection from predators and parasites. This behavior is costly for parents, but beneficial for offspring development and survival. But parents can be selfish. Selfish parents are interested in producing more offspring during their lifetime rather than caring for the offspring. But offspring are not passive recipients of parental care. They actively demand for food and care and influence their parents' behavior and future reproduction. Selfish offspring overrate their own development and survival at the cost of their parents. Instead of maximizing the fitness of one or the other, natural selection favors the well-coordinated parents and offspring. The balance between selfishness and altruism in parents and offspring during their reciprocal interaction is what we call parent-offspring co-adaptation. And the main question in this study is, what is the genetic mechanism underlying parent-offspring co-adaptation? We hypothesized that genes with underlying parent-offspring co-adaptation should have following signatures. First of all, the expression of those genes should alter in parents or offspring during their behavior interaction. And the co-regulated genes with altered expression in both parents and offspring at the same time are the strongest candidate. Secondly, those genes should influence behavior or fitness of focal individual expressing this via direct genetic effects, or influence behavior or fitness of others in the social environment of focal individuals through indirect genetic effect. Last but not least, genes with selfish function are expected to be beneficial to focal individual but harmful to others, and genes with altruistic behavior are expected to be beneficial to others but costly for themselves. In this study, we focused on a non-model organism, the European earwig Formicola auricularia. It's a subsocial species with facultative maternal care. And the earwig mother attended eggs and will groom eggs from time to time. This behavior, as is shown in the video, significantly reduced fungus infection and increased hatching success of the clutch. When the nymphs hatch, earwig mothers provide their offspring with food. And this is the video showing the earwig mother feeding her nymphs mouth to mouth. The communication between earwig mother and offspring is partially through chemical signals such as cuticular hydrocarbon. The earwigs live in groups. Before oviposition, a mated female would leave the group, make her own burrow, and wait for the babies to come. After oviposition, the earwig mother would attend eggs and care for the brood. After the first clutch, the mother either die or will lay a second clutch without further mating. The mother will repeat the care for the second clutch, and the female will eventually die. When the nymphs grow up, they will repeat this cycle again, and there are only 1 or 2 generations each year. The earwig is a great system to study social evolution, as it would fill the gap between solitary and eusocial species. In solitary species here, such as Drosophila, there's no parental care at all. And in eusocial species, such as honeybee, there are more complex social structures and the reproductive division of castes within a colony. Here is our experimental design. We behaviorally manipulated whether or not the earwig mothers would attend the eggs and whether or not they will interact with hatched nymphs in 3 treatments: no care, egg care, and full care. We sequenced transcriptome from 4 tissues of the mother, including antenna, head, abdomen, and ovary, and whole body of the nymphs, using RNA-seq. Candidate genes are identified based on the hypothesized signature of co-adapted genes. Then we characterize the social function of candidate genes using in vivo RNA interference. We predicted that we will get a set of differential expressed genes in mothers between full care and egg care, and a set of differential expressed in offspring. And overlap between mother and offspring are the candidates with co-regulated expression. We found over 1,600 genes in mother and offspring are associated with parental care. This suggests a broadly altered gene expression in parent-offspring interaction. We found 13 genes that are overlapped between mothers and offspring. Among them, there are 2 genes present in 5 other insect genomes, including 2 eusocial species and 3 non-social species. And they are TH and PEP3. One encodes for the rate-limiting enzyme for dopamine synthesis, and the other encodes for an aldrin binding protein. We found upregulation of TH expression in maternal head and offspring during parental care. This suggests the social bond between parent and offspring is maintained by the endogenous dopaminergic reward. And this is further confirmed by the expression of another gene encoding the enzyme in dopamine synthesis pathway AADC that is also upregulated in maternal head in parental care. These results suggest an activated multi-reward function of dopamine in the central nervous system of mothers and offspring during parental care. Interestingly, we found the expression of another gene encoding the enzyme NAT that degrades dopamine is upregulated in full care mothers' ovary, suggesting antagonistic regulation of dopamine in the central nervous system and the reproductive system of caring mothers. And this suppressed gonadotrophic function of dopamine in maternal reproductive system is consistent with the fact that the airway mother suppress their reproductive behavior during parental care. The expression of PAP3 is upregulated in maternal head and offspring during parental care, suggesting an enhanced chemical communication between airway and mother and offspring. And this is further corroborated by the upregulation of multiple desaturations in mother and offspring. Here, desaturations is required for CHC synthesis. And we've mentioned before that CHC is the chemical signal that was used for communication between airway mother and offspring. So far, we have identified 2 genes that are co-regulated in parent and offspring. And our next question is, do they influence behavior and fitness? To answer this question, we manipulated expression of TH and PAP3 in mothers or offspring or both in 3 treatments. And 3 corresponding YFP controls were used to set the baseline for behavior and fitness assay. We used general linear model to disentangle the effect of physical injection expression within and between individuals. Only gene by maternal treatment effect or gene by offspring treatment effect tell us whether we are getting a DGE or IgE. Whether it's a DGE or IgE is based on whether the treatment is in focal individual or others. We predict that we will get behavioral fitness consequence of target gene knockdown in focal individual via direct genetic effect and in others via indirect genetic effect through social interaction. So let's take a look at our results. We found TH knockdown influenced maternal food provisioning. If we compare these 2 groups, we found less mothers provisioning food than corresponding YFP are found in knockdown mothers compared to the untreated mothers, suggesting TH expression in mothers enhanced food provisioning. And next, if we compare these 2 groups, we found fewer mothers than corresponding YFP are showing are investigating a second clutch in mothers with knockdown offspring compared to mothers with untreated mothers, suggesting TH expression in offspring raised the likelihood of maternal future reproduction via social interaction. Next, for TH knockdown, we found a faster development in the offspring relative to corresponding YFP treatment was found in offspring with knockdown mothers comparing to offspring with untreated mothers, suggesting PAP3 in mothers delayed offspring development via social interaction. And next, if we compare these 2 groups, we could see that the mothers showed reduced relative size of second clutch than corresponding YFP control in knockdown mothers compared to untreated mothers, suggesting PAP3 expression in mothers induced maternal investment in future reproduction. And next, if we compare these 2 groups, we found the lower survival rate of offspring than corresponding YFP control in knockdown offspring compared to untreated offspring, suggesting TH expression, suggesting PAP3 expression in offspring increased the offspring's survival. Interestingly, both PAP3 and TH controls maternal future reproduction, and TH expression in offspring regulates whether or not the mother would lay a second clutch through indirect genetic effect, and PAP3 expression in mothers controls how much the mothers would invest in her future reproduction through direct genetic effect. With this, we have shown that both TH and PAP3 influence behavior and fitness. And our next question is, are they selfish gene or altruistic gene? And we found TH is a reciprocally altruistic gene, as its expression in mothers enhanced food provisioning. And this is a behavior that's costly for mothers, but beneficial for the offspring. And its expression in offspring raised the likelihood of maternal future reproduction via indirect genetic effect. And this is the behavior that's beneficial for mother, but potentially costly for the offspring that is expressing this gene. In both cases, this gene is beneficial for others, but costly for the individual expressing this gene. And for PAP3, it's a reciprocally selfish gene, as expression in mothers induced her own relative investment in future reproduction and delayed offspring development. And its expression in offspring increased offspring survival. In all cases, its expression in focal individual is beneficial to themselves, but costly for others. With this, we've shown that PAP3 is a gene promoting selfish behavior in both mother and offspring, while TH is a reciprocally altruistic gene. Interestingly, the dopaminergic pathway appears to be evolutionarily conserved from insect to human. In the earwigs, we found the gene encoding the rate-limiting enzyme for dopamine synthesis is an altruistic gene in both mother and offspring. And in vervet monkey and human, it was reported that dopamine receptor VNTR polymorphism is associated with parental care. On the contrary, the function of PAP3 appears to be co-opted in social evolution. In the solitary Drosophila, its expression in larvae regulates the metamorphosis of the self, and its expression in subsocial earwig mothers regulates the development of the offspring. And it acquires a novel function regarding reproduction, and is regulated through direct genetic effect. If we look at the eusocial termites, there are two copies of PAP3 with caste-specific expression between sterile soldier and reproductive allies, suggesting a potential subfunctionalization of PAP3 in eusocial state. To further understand PAP3 control of reproduction, we hypothesized this molecular pathway through vitellogenin, juvenile hormone, and insulin signaling. And based on our RNA-seq data, we found consistent results suggesting suppressed juvenile hormone and elevated vitellogenin. This antagonistic regulation of JH and VG was once thought to be unique to the eusocial honeybee, but we found the same pattern in the subsocial earwig, suggesting an ancestral function of this pathway in social evolution. To wrap up, we have showed two genes coordinating selfishness and altruism, and our results suggested roles of TH-restricted dopaminergic reward, PAP3-mediated chemical communication, and insulin signaling, juvenile hormone, and vitellogenin interplay in parental care and social evolution. Of course, studies about more parental care species and more organisms covering different social complexities would help with better understanding of parental care and social evolution. With this, I would like to thank all these people and the Swiss National Science Foundation for their support, and all the molecular work was done at Genetic Diversity Center at ETH Zurich. Thank you for your attention.
Thank you, Dr. Wu. So, let's see whether we will have questions first from the advisory board. Do we have questions? Any question? Seems like we do not have questions from the from our experts. So let's see whether we will have questions from the audience. I believe you just gave a really clear talk, so everybody understood you very well. So let's thank you very much again, Dr. Wu, and thank you for this talk. Okay, let's welcome our next speaker, Dr. Sun Huan, who comes from St. Jude Children's Research Hospital, and today she will share her research regarding deep single-cell type protein profiling of mouse brain by non-surgical AAV-mediated proximity labeling. Welcome, Dr. Sun.
Good morning, everyone. Thanks to the organizers. It's my pleasure for sharing our new proteomics technologies. Today, I will present an efficient method which can be achieved deep single-cell type proteome profiling of mouse brain by non-surgery AAV-mediated proximity library. As we all know, central dogma is the gold standard in biology. From 25,000 DNA to about 100,000 RNA and over 100,000 proteins, even to over 1 million protein species, considering post-translational modification (PTMs). We have realized that the complexity increased a lot from the level of genome to the proteome. As executor, proteins play an important role, and the proteome becomes one of the most popular methods to provide protein changes in a large scale. Besides DNA, RNA, protein profiling, combined different omics technology can facilitate a comprehensive understanding of their biological mechanism. To better understand the biological mechanisms, system biology has developed as a new approach to studying the level of organism, tissue, and cell, and putting them together to draw complex interactions with different levels. System biology explores interdisciplinary approaches such as proteomics technology to gain new data and computational analysis and modeling to put out new hypotheses and biological experiment to prove new interactions. Thereby, system biology allows us to study how a phenotype is generated and affected by the genotype, especially with the development of omics technologies. There's large-scale screening and deep coverage speed up the biological interpretation. Recently, tandem mass spec spectrum is a mainstream quantitative proteome. Our lab developed TMT-based LC mass proteomics can handle 18 samples in one experiment. Through isobaric labeling of TMT, 18 samples can be pooled equally and analyzed simultaneously in the mass spectrometry. Isolation isotopic reporterized abundance of proteins can be quantified in 18 samples. After a database search, we can identify and quantify over 10,000 proteins in all samples. What's more, to further increase input, we developed a 27 and 29-plex method combining two sets of TMT reagents, and in sensitivity, we developed micro-scale pipeline, which can steadily report 1 microgram of each sample and identified over 5,000 proteins, which means our proteomics platform achieved a major 7 to 8 order of magnitudes of protein abundance. Quantifying proteins at the book level, like tissues, provide crucial information in many diseases. However, the book proteomics study analyzes the proteome of a mixture of heterogeneous cell types from tissues. The proteins from specific cell types are often masked in the book analysis. Thus, tracing proteins to specific cell type is challenging, especially in brain tissue. Although cell sorting methods, including MAX, FAX, and microfluidic chips, and laser capture dissection can isolate some cell types, but they need unique cell markers to label specific cell types and require specific instrument handling. Besides, during this process, the complex morphology like dendrocyte and exosomes will be damaged and induce the cell death, and functions for synapse or immune cell microglia will be changes. So how to bypass the requirement of cell isolation to gain an in vivo label in specific cell type is a huge challenge. To achieve this aim, we seek the tools to label specific cell type in vivo. One promising tool is proximity labeling by turbo ligands. biotin ligands Turbo ID developed by Elliston Group in Stanford. Turbo ID can biotinate 10 millimeter special proteins in few minutes. Another one is AAV virus for targetable gene delivery to nervous system. We combined these two tools together using their cell type specific promoter to drive the Turbo ID expressions only in neuron to label cell proteins with biotin and using the AAV to deliver the gene expression in brain tissue. Further, through the biotin purification without cell isolation and TMT mass spec, we could identify and quantify the over 10,000 proteins in neurons. To do this, firstly, we confirmed the concept of proximity labeling of entire cellular proteome by Turbo ID. Although proximity labeling was initially developed to probe protein-protein interactions, we reasoned that free Turbo ID can label most proteins in cells. To design this, we construct Turbo ID with flag tag and fused with the T2A self-cleavage peptide, which induced ribosome skipping to translate two proteins independently, and mCherry for imaging. The expression was driven by the cell type specific promoter. As we expected, we observed the Turbo ID diffused in almost all organelles and diffused throughout the cell by immunofluorescence. And then we tested about biotin efficiency of Turbo ID under several conditions. Group 1 without fraction, Group 2 Turbo ID proximity labeling, and Group 3 Turbo ID expression with biotin supplementary. Before real data collection, actually we did a lot of efforts to optimize the biotin purification and subsequent proteome sample preparation. So we managed to identify and quantify a total 10,013 proteins from these samples. In silver staining, compared to very few bands observed in the control samples, many proteins bands are found in the Turbo ID PL samples, and the intensity are significantly enhanced with the biotin supplement, confirming that free Turbo ID is capable of modifying a large number of proteins in cell and comparing the PL biotin groups with the normal cell lysis. The protein mass signature show a strong correlation, which means most biotin labeled proteins had similar protein abundance levels as cell lysis. Reorganizing the potential of Turbo ID-based proximity labeling of cellular proteome, we attempted to develop a strategy for single-cell type protein omics in mice brain. The recombinant AAV was constructed, validated by sequencing, and administered to the mice by retroorbital injection on day 1. After 3 weeks for Turbo ID expression, on day 21, the mice were supplemented with additional biotin for 1 week and harvested on day 28 for imaging and mass spec analysis. We designed KMK2 promoter for neuron labeling and GFAP promoter for astrocyte labeling. In each group, we contained 4 mice with KMK2 or GFAP virus and treated the control group without AAV injection. Using the optimized purification and mass spec workflow, the highly sensitivity protocol led to identification and quantification of 10,294 proteins for less than 5 microgram biotinated proteins. So how to achieve it? Firstly, we validate expression of AAV construction by fluorescence imaging of mCherry, and the expression pattern driven by KMK2 and GFAP promoter are completely distinctive. To further confirm the cell type specific expression, we perform double immune staining of mTERI and cell markers, N for neurons and GFAP for astrocyte. As expected, KIMK2 promoter lead to the recombinant protein expression in N labeled neurons, and GFAP promoter drove the recombinant protein pressed in the astrocytes. In the clustering analysis, AAV effect mouse shows obvious higher intensity than no virus expected mouse. And the sample reproducibility was demonstrated by the results of the replicates. Compared to the negative control, no virus infection, the KIMK2 PL showed the enrichment of 9,919 proteins, and this result indicated a strong effect of turbo ID-mediated biotinylation of brain proteome with high reproducibility. And then we compared the GFAP PL and KIMK2 PL samples to define the cell type specific proteins. A total 672 proteins were decreased in the KIMK2 PL but increased in the GFAP PL brain, which are enriched in the astrocyte cellular components, including cell injection, astrocyte glial cell projection, and phagocytic and endocytic vesicles. Conversely, 1,216 proteins are increased in the KIMK2 PL brain, which are highly enriched the neuron-related cellular components such as synapse, presynapse, postsynapse, dendrocytes, and axon. Conclusion: we reported a novel cell type specific in vivo proteomics labeling approach in which the engineered biotin ligands turbo ID is pressed in the designed cell type in the mice brain using a non-self AAV based strategy. And this turbo ID based approach enabled true profiling of over more than 10,000 proteins from each cell lysate, which markedly improved the proteome coverage, since less than 5,000 proteins are reported in the cell types. The high proteome coverage of our approach is largely competition to the combination of several factors like the robust biotinylation, on-base digestion, highly sensitive protocol, and a newly developed JUMP software. Overall, our investigation provides a solid and effective approach to dissect different cell types in proteome of the mice brain in vivo. What's more, this method is a universal tool to study different cell types using different promoters like inhibitor neurons, microglia, and oligodendrocyte, and is also applied to study specific target interactome like tau, A beta, alpha-synuclein interactome. Further, AAV-based gene delivery can be applied to any animal model without transgenic manipulation. Thus, this tool is a highly potential method of general application in neuroscience research. What we seek is to figure out how a phenotype is generated and affected by the genotype from DNA, RNA, protein to achieve dissecting organelles to tissue and cell. We have developed different level proteome platform from large-scale book proteome like clinical AD human brain profiling to single-cell type proteome like I presented today, and to special temporal proteome like laser capture microdissection of amyloid plaques, even to single-cell proteome, and all four levels could be combined with PTM's proteome like phosphorylation, ubiquitination, methylation. Taking them together, different level of proteomics can further facilitate discovery of molecular mechanism. In the end, I greatly appreciate my supervisor Jimmy Peng's teaching and guidance, and thanks a lot to my lab members and co-members and help. Any suggestions, comments, or questions are welcome. Thank you for your attention.
Thank you, Dr. Sun. Okay, so now I have two questions for you by far, and one is from Dr. Zhang Yan. So the question from Dr. Zhang Yan is, does this only reflect the protein that related to the promoter used to drive turbo ID?
Yeah, thanks for this great question. Actually, the turbo ID expression rely on the promoter. In our method, all the specificity of the promoter decide the expression of turbo. Actually, in here we did the imaging for the turbo ID expression in neuron, and we found the only maybe 50% of neuron, exciting neuron in here, because we use KMK2 promoter. So the exciting neuron, only 50% of exciting neuron express the turbo ID. But we still have the plan to using different promoter. In here, I show inhibitor neuron or oligodendrocyte promoter. We can generate the AAV virus to express the turbo ID in different cell types. Thank you.
Was from our audience. So the next question is from Dr. Zhang Yan. So Dr. Zhang Yan said, could this method be used to dissect different cellular compartments of neuron?
Actually, it can do this because in the Alex Ting turbo ID paper, they use the different like the ER or mitochondria marker to drive the turbo ID expression. So for the neuron, I think if we have the specific promoter, we can drive the turbo ID expressed in synapse, in extrasome or dendrocyte. Yeah, definitely it can do this. Yeah.
And our next question, so it's from Dr. Song Yan. How good this method is to distinguish?
For the distinct neuronal cell types, actually, we plan to this, but we are struggling with how to choose the promoter, how to choose the specific promoter to drive the turbo ID in different neurons types.
Okay, thank you for your answer. Do we have more questions? Okay, so thank you, Dr. Sun.
Thank you.
And thank you for all these questions. Then I will give you our fourth speaker, Dr. Zhang Yunliang. He comes from Brandeis University, and today he will share with us his research about how neuronal structure enhances neuronal robustness to noise perturbations. Welcome.
Hello, everyone. This is Yun Liang from Brandeis University. And today I'm going to talk about how neuronal structure enhances neuronal robustness to noise perturbations. This work is built on the pyloric circuit from the crab stomach. It's a widely used model to study the properties of central pattern generator. And first, let's know about the pyloric rhythm. This is the simplified diagram of the somatogastric nervous system. For the pyloric circuit, it's composed of 4 types of neurons: AB neuron, PD neurons, LP neuron, and PY neurons. These neurons are connected as shown in this simplified diagram. So we need to remember LP neuron receives inhibition from the other neurons. This circuit can spontaneously produce triphasic rhythm. We can see LP neuron, PY neuron, and PD neuron, each of them produce a burst of spikes sequentially. So this pattern repeats at 1 Hertz and is called the pyloric rhythm. We know that for each type of a neuron, they have different ion channels to support the specific spiking patterns. This is the example of PD neuron from the pyloric circuit. We know that although PD neurons from the same or different animals have very similar firing patterns, but they have very different ion channel expression ratios, as shown by the large variations of the mRNA copy numbers that support each type of ion channels. So if we fix all the other parameters and only search the conductances of these channels, then to build models to fit this behavior, then the solution is not unique. Simply speaking, each neuron is different. We still don't know how neurons achieve similar firing patterns when they have these different parameter sets. For some reason, even they achieve it, but the channel conductances are not static. They keep changing for many reasons. For example, ion channels essentially are proteins, and they have a lifespan. If a protein at a dendritic site dies, then the neuron need to sense it, produce, transport, and insert the protein at the same site. But can all these processes be implemented precisely? There's no way. At least these processes will face the spatial and temporal errors. A neural circuit actually can face larger perturbations. For example, neuromodulation is not current specific. It can simultaneously change different components in different directions. For the pyloric circuit, dopamine can change different components. For example, it increases the calcium current in PY neuron, but decrease calcium current in AB neuron. And my work is to explore how a model can still produce similar output patterns when it faces these unpredictable perturbations. So for this work, I use a population of computational models to explore how individual neurons produce to maintain their firing patterns when facing channel conductance variations. I chose the LP neuron from the pyloric circuit. LP neuron receive inhibition from PY neuron and PD neuron. This inhibition triggers rebound bursting in the LP neuron, and this pattern can reflect the pyloric rhythm and be easily classified from the other firing patterns. And therefore, in this work, we focus on this pattern maintenance. So we use 4 compartments, the axon, soma, proximal neurons, and distal neurons, to approximate the LP neuron morphology. Different types of ion channels were inserted in corresponding compartments. That give rise to 14 channel parameters in each model, and there are 1,292 models for the whole model database. So here are the example spike waveforms. The above panel showed the memory potential of the soma, and the bottom panel showed the memory potential of the axon. So the soma and neuron receives inhibition from the PY and PD neurons. And the inhibition triggers rebound slow wave in the soma. The slow wave propagates to the axon to trigger a burst of sodium spikes. But these sodium spikes can only passively propagate to the soma because the sodium channels are only distributed in the axon, but not in other compartments. For the whole population of original models, we call them the parent models. And here I use a simple 2-dimensional schematic to show our perturbation protocols. So for each parent model, we simultaneously change all its channel conductances for M trials. That accordingly, that will generate M child models. And the random perturbation values are sampled according to these formulations. So the child models can produce very different firing patterns, but we only care about how many of them are bursting neurons. So if a model has at least 2 spikes per burst and fire tonically and fire periodically, it's defined as a burster. This is the formulation to calculate the ratio of child models being bursters. It's also used to measure the robustness of a parent model. So here the color codes the robustness value. If the value is 1, that means for this parent model, all its child models can reliably produce rebound bursting, i.e. it's robust to the channel conductance variations. If the value is 0, no child models can produce rebound bursting. It's vulnerable to the channel conductance variations. As shown in this histogram, when the variation range is plus and minus 10%, we can see it's in this histogram, the distribution is significantly right shifted, suggesting that for most parent models, their child models have a high probability to produce rebound bursting. And for 649 of them, actually, all the child models can reliably produce rebound bursting. So when the variation range gets larger from left to right, we can see the distribution is left shifted, suggesting that the neuronal robustness decreases with the variation range. So if we pick 2 groups of neurons that show different robustness levels to the channel conductance variations when the variation range is small, let's say high and low group. So do they still behave similarly when the variation range gets larger? And our simulation results suggest that it's not in this case. Regardless of the initial robustness values, when the variation range is small, we can see all the neurons have similar robustness values when the variation range gets larger. So in previous simulations, we simultaneously changed 14 channel conductances simultaneously. What about if we only change one of them each time? Our simulation results suggest that the neuronal robustness is relatively insensitive to single channel conductance variations, although by comparison, the neuronal robustness is more sensitive to the axonal channel conductance variations. So in the beginning, I have mentioned the soma and neuride receives inhibition from other neurons. The inhibition triggers rebound slow wave on the soma and neuride, and this slow wave propagates to the axon to trigger the sodium spikes. And therefore, we can imagine the coupling between the soma and axon should be critical for the bursting properties and may affect this pattern maintenance. And this is the formulation to calculate the axial resistance. So rho means the axial resistivity. It has the unit of ohm centimeter. And L is the length, S is the area. And in the field of neuroscience, we usually use RA instead of rho. So in this work, I'm going to change the value of RA to manipulate the coupling between the soma and the axon. So a smaller RA means a more coupled soma and axon. A larger RA means a more isolated soma and axon. We should note that in this and the next slide, there will be no channel conductance variations. I only explore how RA or the soma-axon coupling affects the rebound bursting pattern properties. So when RA changes, some parent models no longer produce rebound bursting as shown in this example. So therefore, first we explore how RA affects the population size of bursters. We can see, so when RA is changed around the control value of 100 ohm centimeter, the population size of bursters is relatively insensitive to the RA changes. For example, within the range of 95 and 125 ohm centimeter, all the parent models can reliably produce rebound bursting. And when RA is in the range of 40 to 300 ohm centimeter, over 90% of them can still produce rebound bursting. And only when RA deviates distantly from the control value, the population size of bursters significantly decrease. In this work, we tested RA in the range of 2 to 500 ohm centimeter. We can definitely test it a larger range, but that will significantly reduce the bursting neurons used for the analysis in panel C. So in panel C, we explored how RA affects the number of spikes per burst, i.e. the axonal spiking ability. But as shown in B, some parent models no longer produce rebound bursting when RA changes. So here we chose 130 models that reliably produce rebound bursting irrespective of RA changes. We can see for these models, most of them show biphasic changes with RA, which is also reflected by the population average. However, the number of spikes per burst keep increasing in several neuron models. And next, let's see what's going on. So on the one hand, when the soma and axon are more coupled, the axonal memory potential approaches the somatic slow wave. Here we can see which inactivate sodium channels to reduce axonal spiking ability. A more coupled soma and axon, as reflected by the nearly overlap spike peaks. A more coupled soma will exert a larger capacitance load on the axon to reduce the axonal spiking ability. So therefore, when the soma and axon are strongly coupled, the axonal spiking ability is significantly reduced. If we only consider these two factors, increasing RA that will increase the axonal spiking ability, which is for this example. However, when the soma and axon are more isolated by increasing RA, that will also decrease the axial current from the somatic slow wave to reduce the axonal spiking ability, which is this example. For example, if we go to extreme, we disconnect the soma and axon, then you will never see a burst of sodium spikes on the axon because you lack the axial current to trigger the sodium spikes. Okay, so in previous slides, we have shown you that when RA changes, some parent models no longer produce rebound bursting. And here we chose 131 models that reliably produce rebound bursting when RA is 100 and 500 ohm centimeter. And we explore how RA changes or the soma axon coupling affects neuronal robustness to channel conductance variations. So we can see, irrespective of the variation ranges, so the neuronal robustness, each neuronal robustness with RA being 100 ohm centimeter is always higher than neuronal robustness with RA being 2 ohm centimeter. Similarly, in panel B, we can see, so each neuronal robustness with RA being 100 ohm centimeter is always higher than neuronal robustness with RA being 500 ohm centimeter. So this data suggests that the biological coupling neither too strong nor too weak make neurons robust to the channel conductance variations. So to briefly summarize our conclusions, we found that the rebound bursting in LP neuron models are robust to the random channel conductance variations. The soma-axon coupling determines neuronal robustness to noise perturbations. And thank you, that's it.
Okay, thank you so much. Can you hear me? Oh, thank you so much, Dr. Zhang. And we have one question from the advisory board. And in this model, do you consider the capacity of the cells, for example, the diameter of axon or area or volume of soma? It's from Dr. Zhang Yan.
Thank you for the question. Yes, I mean, these models are, although they are simple actually, but we still try to replicate the real morphology. So, I mean, the reason we did this work is previously people usually explore neuronal robustness by single compartment models. That means they squeeze everything together using like a single ball, and then they build the models and then explore how these kind of models are robust to the channel conductance variation. Of course, it sucks. And in this work, we try to include different compartments, like put the slow waves, the kind of signal on the soma and neuron, and the spike initiation side in the axon. So the separation of different signals make the neuronal bursting patterns robust to the variations. That's basically our story.
Thank you so much. And we have more questions from the audience or from the advisory board. I think you just got everybody clear. And thank you so much, Dr. Zhang. So the first session of our forum is now over, and let's have a short break. So remember to come back at 11:15. Thank you very much. Thank you for everybody's patience and time. Thank you. Welcome back from the break, everyone. Let's continue enjoying our excellent reports. I'm the hostess of the second part of the forum. My name is Luo Peiyi, a postdoc from Cyber. In the second part, we have 3 presentations ahead. Each presentation will take 15 minutes for the speaker and 5 minutes for us to ask questions. The first question will be from our advisory board. Now, here comes a postdoc from Noah Palm's group in Yale University. The report she's giving today is entitled Human Commensal Bacteria Produce Novel Genotoxic Metabolites that Exacerbate Colorectal Cancer. Let's welcome Tao Yiyun.
Hi, everyone. Many thanks for the very kind introduction. It's my honor to have the opportunity to share this story at such a interdisciplinary forum. Firstly, I'd like to start with a brief introduction of the microbiota, a word you may hear a lot in the past decade. The humans are consistently colonized by trillions of microorganisms, including bacteria, fungi, viruses, and parasites. They are distributed at all mucosal and barrier surfaces, including nasal cavity, oral cavity, lung, skin, gut, and vagina. Particularly, the GI tract contains the largest number of microorganisms and harbor over 10 to the 14th power of bacterial cells from multiple phyla. Besides intradiversity, the microbiota also shows inter-individual diversity associated with sex, age, and environment. Many groups already show that alterations in microbiota, which is also called dysbiosis, are associated with diverse disease states, both locally and systematically. In GI tract, inflammatory disease like IBD are widely studied, and other disorders like IBS, food allergy, and cancer are also related to the gut microbiota dysbiosis. The gut microbiota has distal effects on other organs or tissues, found to affect feeding behaviors, neurodegenerative disease, metabolic disease, and autoimmune disease. However, the correlation doesn't mean causation. Compared to the human, the gut microbiota contains about 154 genes and thousands of metabolites at the individual level, and even more in population level. The majority of these microbial genes and products lack functional annotations. Today, I will show how we investigate the causative role of the microbiota in colorectal cancer. CRC is the third most common malignancy and the second leading cause of cancer deaths worldwide. 2/3 of all CRC cases occur in individuals without a family history of CRC or inherited CRC predisposing genetic mutations. The environmental risk factors that promote the acquisition and accumulation of somatic, genetic, and epigenetic aberrations are chief contributors to CRC development. The gut microbiota has been well described as a significant factor in CRC, and there are diverse mechanisms for it. For example, short-chain fatty acids can modulate immune responses in the tumor microenvironment. Direct host microinteractions could change the proliferation signaling pathways in intestinal epithelial cells, such as Wnt beta-catenin signaling. Microbial products can also trigger DNA modification in intestinal epithelial cells. Besides indirect DNA damage due to the ROS or RNS, direct bacteria-inducing genotoxicity is also observed, like cytolysal distending toxin from pathogenic gram-negative bacteria has directed DNase activity. More interestingly, a small molecule metabolite colibactin also influences CRC risk by directly causing DNA damage. The colibactin biosynthetic machinery is encoded by a 54 kb hybrid PKS-NRPS gene cluster referred to as the PKS-CLB locus. This locus is prevalent in IBD or CRC patients, indicating the correlation between colibactin-producing bacteria with human CRC, which was confirmed by PKS-positive E. coli-induced tumor progression in mouse models. The mature chemical structure of colibactin, responsible for the DNA interstrain cross-linking activity, was recently determined by 2 biochemical labs at Yale, who are also our collaborators in this project. Furthermore, human CRC tumors also contain mutational signatures consistent with colibactin-induced DNA damage, implicating colibactin in human CRC development. However, aside from colibactin, the role of microbiota-derived small molecule genotoxins in CRC initiation or progression remains mostly unexplored. Given the enormous complexity and diversity of metabolites produced by bacteria, we hypothesize that previously undiscovered small molecule genotoxins produced by diverse gut microbial species also contribute to CRC. To test this, we established a pipeline to screen a gut microbiota culture collection consisting of 122 isolates. We co-incubate individual bacterial isolates with linearized plasmid DNA based on the principle that the extent and modes of DNA damage can be assessed by electrophoresis and native and denaturing conditions. So, the double-strand linearized DNA migrates as a slow-moving band under native electrophoresis, while denaturing treatment separates the DNA into single-strand DNA, leading to a hair mobility band. The formation of DNA interstrand crosslinks, for example by colibactin, prevents unwinding under denaturing conditions. Accumulation of DNase-mediated degradation can result in damaged DNA products detected as smaller fragments, and DNA damage induced by restriction enzyme-like molecules produces multiple bands. Here are 18 putative genotoxic hits we screened based on this electrophoresis assay. Unlike the interstrand crosslinks induced by colibactin-producing E. coli strains, most of these candidate isolates exhibited accumulation or DNase-like patterns. Some isolates also combine with restriction enzyme-like patterns, with the DNA cross-linking chemical cisplatin as positive control. We found that for isolates from Clostridium perfringens, Clostridium ramosum, which is phylogenetically related to C. perfringens, and 2 isolates of Morganella morgani induced increased gamma H2AX, which is a marker of DNA double-strand breaks with their small molecule supernatants in HeLa cells. Notably, morgani has significantly increased abundance in IBD patients, specifically in Crohn's disease patients in human microbial project data, suggesting potential roles in IBD patients who are at an increased risk of CRC diagnosis and death. Therefore, we prioritize morgani for genotoxin identification. To do this, we employed the QTOF-MS-based untargeted metabolomics to generate a morgani metabolite ion list, and use bioactivity-guided fractionation to exclude inactive ions. Then we performed a large-scale cultivation of mahogany to acquire fractions enriched in potential genotoxic N features, and using NMR to identify novel chemical structures. We found that fraction F2 exhibited genotoxicity in plasmid-based DNA damage assay, and it was primarily comprised of two previously undescribed metabolites. Compound 1, a target N feature from metabolomics, contains an imine group, which is a carbon-nitrogen double bond. Thus, we termed this novel metabolite indolamine 214. We synthesized these two compounds and confirmed that indolamine 214, but not compound 2, triggered increased gamma H2AX in a dose-dependent manner. In addition, indolamine 214 was just detected in bacterial cultures of mahogany instead of Colibactin-producing or non-producing E. coli strains, and it's just present in mahogany colonized mice. Moreover, we also discovered two additional genotoxic indolamines, indolamine 200 and indolamine 248. They were also detected in mahogany bacterial cultures and mahogany colonized mice. The imine group is likely derived from the spontaneous condensation of a primary amine and the aldehyde of indole-3 aldehyde. Primary amines are microbial metabolites that can be synthesized from amino acids via a one-step reaction mediated by bacterial decarboxylysis. Through a genome-wide transposon mutagenesis campaign involving the generation and mapping of 16,000 mutants, we constructed an AAT isogenic mutant that failed to produce indolamines. Then we used this isogenic mutant strain to evaluate the impacts of mahogany-derived indolamines on CRC. We colonized non-biotic mice with AAT positive mahogany or AAT negative mahogany in the context of a non-genotoxic mock community, and then treated with AOM and 3 cycles of DSS to induce CRC. We also used Colibactin-producing or its mutant as a pair of controls. As expected, colonization with CLBP-positive E. coli NC11 promoted colorectal tumors as compared to CLBP-negative E. coli NC11. Similarly, mice colonized with AAT-positive mahogany also exhibited increased tumor numbers and tumor scores as compared to mice colonized with AAT-negative mahogany. Moreover, mice colonized with AAT-positive mahogany exhibited an increased ratio of adenomatous lesions with high-grade dysplasia per tissue section, indicating accelerated tumor progression. Finally, we mined the cancer microbial database and found that mahogany exhibited increased prevalence or abundance in primary GI tumors, including colon tumor, rectum tumor, and stomach tumor, as compared to multiple non-GI tumors. Mognella was also significantly enriched in tumors from rectum and the stomach as compared to adjacent solid normal tissues. Notably, we also found that AAT gene was conserved across nearly all mahogany strains in the NCBI database, suggesting that indolamine production may be a conserved feature of mahogany. Overall, our data suggests that genotoxic indolamines from mahogany may serve as tumor-inducing factors in humans. In summary, I hope I show you how we dissect the causative effects of human commensal metabolites on CRC. We undertook a systematic evaluation of the genotoxic potential of human gut microbes, identified and characterized a novel family of genotoxins indolamines and their responsible gene. We also confirmed indolamines' effect on colon tumorigenesis in vivo with isogenic mutants. This work is a very collaborative work from our Palm lab, Jason Crawford lab, and Seth Herson lab. We also got lots of help from Dr. Wenju Hu, Jiawei Wang, and the Yale Center for Molecular Discovery. We also got help from Yale Animal Resources Center for Germ-Free Mice and the Yale Center for Genome Analysis. And many thanks to the CRC scholarship to support my PhD and all PalmLab members to help with my experiments. Now I'm happy to take questions. And if you have more questions, also feel free to send an email to the y.cao@yale.edu, and I will try my best to answer all of them. Thanks. Hi.
Many thanks to Yiyun for giving us such an impressive talk. The first question will be from Advisory Board. Now we have a question from Dr. Chen Jian. Do CRC patients with M-mogni have higher mutation load comparing to M-mogni negative population?
Many thanks to the question. That's a really great question. So it's very interesting that actually it really depends on if we can have like more public data set, or if we have more CRC patient samples to analyze this. We'd like to see that, but unfortunately we don't have the human samples to that. So what I just show is that we can, using the public data set, to mine the microbial abundance, or we can try to observe the prevalence of the bacteria. So we'd like to see related research in the future, but I have to say now I don't have an answer for this question. Thank you.
Thank you for the answering, and thank you for the question. Thank you for the question from Dr. Chen. And seems like we don't have any questions from the audience, so we only have these questions for Yiyun. Thanks again. Oh, there is another question coming out. Wait 1 minute. It's from Dr. Chen. How about the mutation load in your mouse tumor model?
Yeah, that's also a great question. We haven't analyzed that. So what we just show is the overall tumor load and the tumor, adenoma in this experiment, but we haven't do the whole genome sequencing to measure the overall mutation load. Yeah, but it's a very good suggestion, and I would like to try that with our tumor samples. Thank you.
Okay, good afternoon, everyone. Welcome back to the meeting. My name is Han Yu, and I am a postdoc in the Professor Chen Peng's lab at Peking University. I will be the co-host in the panel this afternoon. We have 11 talks on the agenda, each of which contains 15-minute report and a 5-minute discussion. A 10-minute break will be given after the fourth one. Now, let's get started. I am pleased to invite our first speaker, Dr. He Dan from UC Berkeley, to give the talk about the chemoproteomics. Her topic is methionine oxidation activates pyruvate kinase M2 to promote pancreatic cancer metastasis. Let's welcome Dr. He.
Hello everyone, thanks for having me here and thanks for the nice introductions. My research interest is protein chemical biology, where I use chemoproteomic strategies to study biological questions related with protein post-translational modifications and protein-protein interactions. The work I'm going to present today is at the interface of redox biology and chemoproteomics. So questions we are interested in is whether antioxidation prevents or promotes cancer. Although reactive oxygen species, ROS, have been implicated in tumor development, the fundamental relationships between redox biology and cancer remain poorly understood. In addition to serving as indiscriminate oxidants, some ROS, such as hydrogen peroxide, can regulate redox signaling through reversible oxidative post-translational modifications on cysteine residues. For example, redox signaling through specific cysteines has been implicated in PDA development. The pathogenic mutations of KRAS oncogenes induce NERV2, a transcription factor, and elevates the levels of endogenous antioxidants to maintain the reduced state of selected cysteine sites within multiple translational machinery to sustain primary tumor growth. This example demonstrates more sophisticated redox signaling mechanisms beyond indiscriminate oxidative damage in primary PDA tumor growth. However, it remains unclear whether distinct redox signaling mechanism also contribute to the high metastatic potential of PDA. On the other hand, whereas cysteine has been well recognized as functional redox switches, less attention has been focused on methionine, the other sulfur-containing amino acids, and its reversible oxidation to methionine sulfoxide. The reduction of methionine sulfoxide is dependent on an enzyme called methionine sulfoxide reductase. It has A form and B form. It's unclear that whether methionine residues can also serve as physiological redox switches that cycle between the oxidative and reduced states to regulate specific biological processes such as tumor development. To address those questions, we did immunohistochemical analysis of tissues from patient-derived pancreatic, normal primary tumor, and secondary liver metastasis. The results reveal that the methionine sulfoxide reductase A (MSRA) expression is reduced in primary tumor cells compared with adjacent normal tissues and almost undetectable in liver metastasis. This expression pattern was also observed in the immunoblot analysis of a mouse pancreatic organoids model, and whereas the MSRB1 and B2 express comparable levels during the PDA development. And PDA-T here represents the tumor settings of PDA, and PDA-M means the metastatic settings of PDA. The genetic ablation of MSRA provides no growth advantages to PDA T cells in vitro or in vivo upon orthotopic transplantation. Strikingly, however, we observe a substantial increase in the incidence of metastasis upon MSRA deletion. Interestingly, the PDA organoids with deletion of MSRA migrate through the microgel and give rise to the 2D model layer outgrowth, whereas the controls maintain the 3D growth. And we also observe an increase of cell migration in the wound healing assays upon MSRA deletion, whereas ectopic expression of MSRA suppressed migration. The PDA cells with MSR deletion also phenocopy the migration of PDA-M cells, whereas MSR deletion in PDA-M cells does not further accelerate migration, presumably because basal MSRA expression in PDA-M cells is already very low. Consistent with these observations, we found that MSR-deficient PDA T cells colonize the liver more efficiently than MSR-expressing controls upon intrasplenic injection. To further examine the tumor suppressive functions of MSRA, we generate PDA-M cells to express MSRA in a doxycycline-inducible manner. MSRA induction significantly reduces liver colonization when injected intraspinalically. Notably, whereas secondary macromads were observed in every mouse of the cohort, none were observed in mice administered with doxycycline. As the oxidation of free and protein-bound methionine has been shown to scavenge ROS, we ask whether the downregulation of the MSRA results in elevated global levels of ROS, which in turn might promote PDA metastasis. And we did a thorough analysis of the global ROS and also the growth factor signaling pathways, and now it's showing a dependence of MSRA. Given these observations, we hypothesize that MSRA suppresses PDA's metastasis by controlling the oxidation state and thus function of a selected group of protein targets. To identify the protein targets of methionine oxidation that underlie PDA metastasis, we use our chemoselective bioconjugation approach termed REACT, which relies on oxyzerdine warhead that can specifically label methionine in the proteome. We coupled REACT with quantitative proteomics to profile the reactive methionine proteome of pancreatic organoids. Here in methionine, oxidation is defined by the loss of reactivity towards oxyzerdine alkyne. By normalizing reactive methionine changes across the proteome, we were able to identify methionine oxidation events independent of protein expression differences. And we find a significant decrease in protein-specific methionine reactivity reflect an increase in methionine oxidation from normal to tumor to metastasis, with the most robust changes observed in the metastasis settings. This pattern of methionine oxidation is in great agreement with the pattern of MSRA expression. To determine whether the subsets of methionine oxidized proteins represent particular biochemical process, our datasets were subject to catch a pathway analysis, and we found that methionine oxidized proteins are most highly enriched for participation in metabolic pathways. And more than half of the methionine oxidized proteins are involved in central carbon metabolism. These results suggest that cyclic oxidation and reduction of the methionine residues on select proteins may control the glucose metabolism in metastatic pancreatic cancer. To examine the function downstream of such oxidative modifications, we perform a seahorse analysis on a panel of PDA T cells that are either proficient or deficient for MSRA. The deletion of MSRA does not alter glycolysis as measured by extracellular acidification rate. However, we saw an increased mitochondrial oxidative phosphorylation as measured by oxygen consumption rate when MSRA is deleted. Upon heavy isotope tracing of uniformly labeled C13 glucose, we observed a marked increase in pyruvate, but not its proximal glycolytic intermediates. In the MSRA deleted PDA T cells, and all downstream metabolites of the tricarboxylic cycle display elevated C13 labeling, confirming that MSRA loss promotes glucose oxidation. Moreover, this increase in pyruvate and TCA cycle intermediates labeling was also observed in the PDA-M cells. Accordingly, MSRA-deleted PDA T cells and PDA-M cells generate more ATP from respiration than the control PDA T cells. As ATP is a main energy currency of the cell, elevated ATP synthesis likely serves to power the bioenergetics of cell aggregation. In accordance with metabolic tracer analysis indicate that glucose oxidation is increased as the step from PEP conversion to pyruvate. Our chemoproteomic profiling also confirmed that this enzyme PKM is strongly oxidized. Then we wanted to study the biochemical regulation of PKM by MSRA. The increase of PKM methionine oxidation and enzyme activity is both seen in PDA M cells versus PDA T cells and also in PDA T cells upon MSRA deletion. Indeed, the mammalian PKM gene encodes two alternatively spliced products, PKM1 and PKM2, with very different enzymatic properties. To evaluate the impact of MSRA on PKM1 versus PKM2, each protein was ectopically expressed in PDA T cells in the background of plus and minus MSRA. As shown here, immunopurified PKM2 but not PKM1 display increased pyruvate kinase activity, and further, the oxyzerine alkyne labeling confirms that the PKM2 is increased. The methionine oxidation does happen on PKM2. There are 16 methionine residues on PKM2. Based on our chemoproteomics profiling, M239 is most strongly oxidized and it locates to allosteric regulation hub. Importantly, the replacement of M239 with a redox insensitive leucine abolished the induction of PKM2 activity in MSRA-deleted PDA. PKM2 is known to regulate its activity by switching between low-activity dimeric and high-activity tetrameric states, allowing cellular metabolism to shift between glycolysis and mitochondrial respiration, respectively. Using size-occlusion chromatography to examine the molecular weight distribution of PKM2, we found that PDA T cells deficient of MSRA enrich for high molecular weight of PKM2 wild-type protein, but not the mutant protein. We then ask whether pharmacological activation of PKM2 can promote cancer metastasis and phenocopy the loss of MSRA. We use a small molecule that can specifically activate PKM2, and by doing so, we observe that the small molecule treatment groups showing a significant increase in liver colonization, which suggests that the pharmacological activation of PKM2 can promote the metastatic spread of pancreatic cancer. To summarize, here we show that MSRA is a suppressor of pancreatic cancer metastasis. Its loss remodels glucose metabolism by allowing oxidation of a methionine residue that allosterically regulates PKM2. This methionine-dependent redox switch promotes mitochondrial respiration, migration, and metastasis. Finally, I'd like to thank my lab at UC Berkeley, my collaboration CHIO lab in Columbia University, and my fellowship, and all your attention here. I'm happy to take any questions. Thank you.
Okay, time for discussion. Any questions or comments from our advisory board? Okay, I have a question. I wonder how to link the results of Bmpfc and the pancreatic cancer metastasis.
Yeah. Thank you for your questions. So first thing is we saw the enzymes that regulate methionine oxidation, or we say the reductase, is decreased. And then for the ABPP, we can do a statistic analysis to see which one is significantly oxidized. And then we did the pathway analysis and saying that seems like the glucose metabolism is involved in the cancer metastasis. And then we did tracing analysis and combined the ABPP results and our phenotype and the tracing results. We figure out the pyruvate kinase might be the targets we should go for. Then we did more biochemical studies on that proteins to confirm that it's our real targets.
Okay. A question from Dr. Li Yongzheng. Could you explain again how the oxidation was detected?
Oh, that's a great question. So actually, it's like it's kind of like a hypothesis, or our methionine oxidation is defined as a loss of labeling towards the probes. So we hypothesize that once the methionine become oxidized, it cannot react with the probe, since the probe can only specifically label the reduced form of methionine. So then we can do an analysis on the loss of the labeling, and then to predict that is the increase of a methionine oxidation. So because we don't have a specific probe for methionine sulfoxide, so we can only detect the loss of signal by labeling. Mm-hmm. Thank you.
Thanks. More questions from the audience? Okay. Thank you again, Dr. He.
Thank you for having me here. Thank you.
Dr. Li Yongjun, his studies are focused on the current pandemic, COVID-19. The topic of his lecture is T-cell responses to SARS-CoV-2 Omicron spike epitopes with mutations after the third booster dose of an inactivated vaccine. Now, let's welcome Dr. Li.
Good afternoon, my friends. Thanks for the invitation. It's my honor to share my work at this international conference. I am Li Yongzheng from Peking University. My report focuses on T-cell responses and Omicron mutations. I noticed that my report does not fall well with our topic, the brain, and may not be interesting to most of my audience. So I will make my report as simple as possible. You know, the COVID-19 epidemic does affect our life. The emergence of the Omicron variants has raised an alarm related to the control of the disease. I hope after my talk, when you are questioned by families and friends that whether and how the Omicron variants would change our immune responses, you may feel easy to answer them. With limited time, please allow me to briefly introduce my supervisors. I got my PhD degree in Sun Yunjie's lab in Peking University. Professor Sun's lab focuses on the development of super-resolution imaging methods and the investigation of biological processes in the nucleus. In his lab, I worked on the dynamics of the chromatin and DNA replication. In the year of my graduation, the COVID-19 began and spread quickly over the world. I wanted to do something in the fighting against COVID-19, so I joined Sunny's lab and coordinated with Professor Su Bin. We worked together and investigated the effects of Omicron mutations on T-cell responses. As a background, firstly, I want to talk about the effects of Omicron mutations on humoral and cellular immune responses. Indeed, we can get conclusions from the titles of these excellent works. When we refer to antibody, the word widely used is escape. This is because neutralizing antibodies recognize the structure of the whole antigenic protein, which would be changed by the mutations and thus lead to antibody evasion. But when we refer to T-cell responses, the word widely used is cross-recognize, because the T-cell responses to the Omicron variants is based on dozens of peptide epitopes. Even for the Omicron strain with more than 30 mutations, about 80% of the antigenic peptides recognized by T cells are still identical to those of the original ancestral strain. Therefore, T cell responses are largely preserved. Then, as the second part of the background, I want to discuss about the drawbacks of previous reports. The classic method to investigate T cell responses is like this. Firstly, collect the Bmpfcs of the volunteers. In the culture, antigenic peptides were added. Then the antigens are presented to the T cells, and the T cells are activated. Then T cell responses could be detected by related assays like this. We should note that in previous works, peptides with and without mutations are mixed together and used together for T cell stimulation. So the impacts of mutations on T cells might be underestimated, which brings three problems. The first is the underestimation of accumulation of mutations. If mutations lead to epitope escape from T cells, the observation that T cell responses are largely preserved could be the result of the limited accumulation of mutations. After enrichment with more mutations, new variants may completely escape the immune system in the vaccinated individuals. In this case, vaccines focusing on the mutations of variants are needed. The second is the overlook of individual differences. If some mutations allow the virus to escape the T cell responses, a distinct proportion of individuals with certain kind of human HLA types that tend to recognize the viral peptides with mutations may be more susceptible to the impact of the Omicron variant. In fact, reduced T cell activity was recently reported in about 20% of the participants. The third is the estimation of new variants. Identification of the mutations that can reduce T cell responses induced by vaccines may provide clues to evaluate the spread and virulence of new emergent Omicron variants. So it is important to specifically study effects of different mutations. To do this, peptides with and without mutations are used separately for the T cell stimulation. Samples were collected at week 0, week 2, and week 4. Week 0 means the time 6 months after when the participants were vaccinated with the second dose, but before receiving the third booster dose. The notations week 2 and week 4 denote the length of the time after the third booster dose in the same group of participants. Our data reveal that booster vaccination substantially enhanced T cell activation in response to both the ancestral and Omicron epitopes, although the enhancement was slightly weakened by the Omicron mutations. To further investigate which mutation in the Omicron spike proteins evades the T cell responses to the epitopes, 8 peptides with 17 mutations were used separately to stimulate the Bmpfcs collected at week 0 and week 4. Interestingly, compared with the ancestral peptides, only the peptides with the G339D or N440K mutations change T cell responses. To further assess the impacts of G339D and N440K mutations on T cell responses, cytokines in the superintendents of the wells assessed in the ELISpot assay were detected by the multiplex cytokine detection assay. Beads are like this. There are different kinds of beads with different antibodies. then the different antibodies will capture the different analytes cytokines in our assay, like this. And then another antibody with biotin will be bound again by the analytes, like this. So this is the structure like the sandwiches, the bead with the antibody, and then the analyte, and then the antibody with biotin. Then PE and stratividin will be added, so the color will be added to the beads. Indeed, the beads are differentiated by size and internal fluorescence, APC, like this. So we can separate the beads and know which beads will detect what kind of analytes. And then the analytes will be analyzed by PE, the color of PE. So at last, the concentration of a particular analyte is determined using a standard curve generated in the same assay, like this. So this assay is good because it has high throughput and high sensitivity. By multiplex cytokine detection, we discovered that G339D or N440K mutations reduces T-cell responses with decreased expression of IL-6 and TNF-alpha. So these are take-home messages. In the reports from our lab and other labs, data reveal that Omicron escapes the majority of existing SARS-CoV-2 neutralizing antibodies, while T-cell responses to SARS-CoV-2 spike cross-recognize Omicron, and booster vaccination substantially enhanced T-cell activation in response to both the ancestral and Omicron epitopes, although the enhancement was slightly weakened by the Omicron mutations. For our Chinese vaccinees, G339D and N440K mutations are very important. I thank Sunny for his support, and I thank my collaborators from Professor Su Bing's group. Thanks, and questions?
Okay, thank you, Dr. Li Yongzhen. Okay. Any questions or comments from our advisory board?
Maybe, can you hear me? Maybe I should answer the question from Professor Li. Thanks, Professor Li. Indeed, we next checked for the vaccinees, and we also investigate the mutations, Omicron mutations, to the vaccines in people living with HIVs. So we found that it is interesting that we found after the booster dose, cells marked by CD38 and HLA-DR are activated, but the activated cells do not correlate with any kind of cytokines. In that case, we don't know some activated cells and their function. Indeed, it is very interesting. That means when we got the booster dose of the vaccination, we can find some T cells are activated, but we don't know the function of them. So we want to explain the functions of many different kind of activated cells.
Okay, another question from Dr. Zhou Li. Are the mutations of Omicron test very popular right now? How about forced booster of vaccine?
The mutations, yes, about 10 kinds of mutations are proved to be very important, including the G339D and N440K mutations, and some mutations like S371 like this. Yes, there are many important mutations, and for the booster dose of vaccine, I think you see, for a long time, our vaccines, the efficacy of our vaccines may decrease slowly. And maybe there should be the fourth booster dose. I don't know that because this is the policy. I can't I can't tell about the comments.
Okay, thank you, Dr. Li. I have an additional question about based on your studies, do T cells play a more significant role than the B cell upon vaccination administration?
It is hard to say which is more important. I think in for the long time, we can see that the memory B cell and the antibody will decrease, but T cell responses will maintain a far more long time. At the early time of the COVID-19, research published on Nature stated that even after 17 years, the T cells for the SARS-CoV-1, the SARS-CoV-1 in the 2003, the T cell responses still remain. In that case, T cell responses could remain a longer time, but T cell responses, the function of the defending to the virus for T cells is not very direct. We can see that B cells they can secrete the antibodies, so the B cells play a more direct and role to the defense of the virus. So they are not different. So it is not easy to say which is more important.
Okay. Thank you again, Dr. Li.
Thanks.
No. Any more questions from the audience? Okay. If there are no more questions, let's take a brief break and please come back before 50:30. See you later. Bye.
Hello. Okay, I hope everyone has returned from the break, refreshed and ready to go because we have quite the eventful second half. But before that, let me briefly introduce myself. My name is Emily Han. I am a postdoc in Dr. Joji Tsunada's lab at Cyber, and I'll be your moderator for the remainder of this panel today. A refresher for today's rules. For the rest of this panel, we have 7 presentations coming. Each presentation consists of a 15-minute presentation, and after that, we'll have 5 minutes for questions from the advisory board as well as the audience. Now, without further ado, let's welcome Dr. Wang Lun from Peking University. His work focuses on the neural circuit mechanisms underlying neurodynamics and behaviors, especially using live neural imaging techniques. The topic of this talk is cocaine induces locomotor sensitization through a dopamine-dependent prefrontal cortical pathway. Dr. Wang Lun, the floor is yours.
Hi, good afternoon, everyone. I'm Lun Wang from Peking University, and my advisor is Professor Zhuan Zhou. My presentation title is Cocaine Induces Locomotor Sensitization Through a Dopamine-Dependent Cortical Pathway. In this research, we study how prefrontal cortex responds to acute cocaine, and we found a new circuit here. We know cocaine is an addictive stimulant that ranks only after heroin, as there are green arrows indicated here, from the 2 dimensions, physical harm and dependence. In history, cocaine is one of the most popular traditional drugs. It has also been widely used for the study of drug abuse and many other brain functions. In brain, we know for normal synaptic transmission or volume transmission, transmitters and modulators are released by presynapse and recycled by the transporters, for example, dopamine transporter DAT. However, Cocaine is a drug that can target the dopamine transporter and block the reuptake of dopamine. This will produce enhanced dopamine signaling between pre- and post-synapse. If repeated using cocaine, the synapse and the neural circuits will be changed and addiction may happen. And we also need to know cocaine is a drug with multiple targets like noradrenaline transporter NET and serotonin transporter SERT. They have similar binding affinity with cocaine. And for cocaine and almost all drugs of abuse, the most famous circuit is dopamine projections from ventral tegmental area outside VGA to nucleus accumbens NAC. They are related with reward, motivation, and locomotion, and many other functions. However, in recent years, the prefrontal cortex (PFC) is also reported to be related with compulsive cocaine seeking. As a central part of the brain, our question is, at the very beginning of drug exposure, how does PFC get involved? We checked the literature and we found that there's no direct answer to this. So we propose these questions. How does acute cocaine regulate PFC? What's their function and what's their mechanism? We first chose the subregion of PFC frontal association cortex, FRA, to observe the cocaine effect. We first use a custom dye, calibrate 5-0 to monitor the activity. And we define the spontaneous somatic custom signal as SSCS, and we use it to quantify the neuronal activity. Here is the sample traces for neurons recorded in awake mice. The left black traces is control group in the left, and the right red traces are for cocaine group. And we found the percentage of silent cells increased from about 5% by saline and to 30% by cocaine. Is really dramatic or robust phenomenon. And the average SSCS frequency is decreased about 50%. The right lower panel is the cumulative distribution of the neuronal activity. It shows the cocaine-induced change. However, the calcium dye is not cell type specific, so we use virus mediated expression of CAMP in excitatory neurons. And we found the similar inhibition effect is also about 50%. Maybe it's because the excitatory neurons is about 80 or 90% of the prefrontal cortex neurons. And we also observed the inhibitory GABAergic neurons, but we didn't observe any significant effect for GABAergic neurons, as the statistical result shown in the right panel. So we get a short summary that cocaine inhibits excitatory neurons of FRA in awake mice. Then what's the function of cocaine inhibition of FRA? We designed a strategy that use chemogenetic tool HM3DQ to increase the firing of neurons to prevent the cocaine inhibition of FRA. And indeed, we found it can prevent the cocaine effect. It was confirmed by in vivo imaging and slice electrophysiology. Then we combine it with the behavior test. First is traditional or is conditioned place preference, CPP, is a kind of reward memory test for cocaine. As shown in the left lower picture, the left side box paired with cocaine will be preferred by mice, and the red trace will be denser than the right. However, for the saline conditioned mice, they have no preference. And the cocaine conditioned mice will have a big CPP score to the conditioned box. However, compared to the control group, we found no significant loss of function by CPP score. That means the FRA may not be necessary for the reward memory. Then we tested in the locomotor sensitization. Cocaine can make the mice hyperactive, and sensitization means the same cocaine dosage in consecutive days can induce larger and larger stimulating effect of traveling distance. And in the red panel, the upper black line is the control group with normal sensitization. The distance induced by same cocaine was increased in day 2 and day 3 compared to day 1. However, in HMCDQ mice, the upper red line, we found the cocaine sensitization is abolished. So we found that FRA is at least required for locomotor sensitization here. Then we are going to study the mechanism. We want to know what's the mechanism from the molecular and cellular level. We know the cocaine injection was intraperitoneal injection, and in theory, it can reach the whole brain and target any receptor and affect any circuit. So we first utilized pharmacological tools to specifically mimic the blocking effect of each transporter, include GAT, NET, and SERT. And we found only the drugs targeting DAT like normoxensin and GBR. They can produce similar inhibiting effects, but not for NET or SERT. So we conclude that blocking DAT is sufficient to inhibit FRA. Then we use the genetic model to further confirm the role of DAT. DAT knockout mice and cocaine-insensitive mutation DAT-CI mice. Compared to the wild-type mice, they both have no significant response to cocaine. So we conclude that DAT is necessary for cocaine inhibition of FRA. Now the molecular target was confirmed, but dopamine neurons project to a lot of areas. Which pathways can mediate their effect? We first depleted the dopamine terminals in FRA by the strategy to local injection of 6-hydroxydopamine. Below is the immunofluorescence image of anti-TH antibody, showing the depletion effect of 6-hydroxydopamine in FRA. And we found in the right upper panel, after 6-hydroxydopamine depletion, cocaine can still inhibit the activity with similar effect. So this means the direct projection is not responsible for the effect. Then which pathway? We design another strategy to screen. We first find from the upstream of FRA, and we use retrograde AAV to identify the upstream areas of FRA, since FRA is a new brain areas with relative small number of literatures. And we found many afferent areas in the whole brain, like insular cortex, Bmpfc, amygdala, and the parental cortex. Among them, the BLA and the Bmpfc receive well-known dopaminergic projections. These upstream areas with dopamine innervation could be potential targets, we believe, or we guess. So we use the same strategy to deplete the dopamine terminals in BLA and Bmpfc. We show here the BLA basolateral amygdala dopamine was not necessary for cocaine inhibition of FRA. However, The dopamine depletion in Bmpfc showed a significant abolition of cocaine inhibition. The right panel is the statistical result showing the loss of function. Thus, we screened and we found Bmpfc dopamine-mediated cocaine inhibition of FRA. Bmpfc is important. Then, for the details of this projection from Bmpfc to FRA, we use interrogate transsynaptic virus AAV1 to label the postsynaptic neurons in FRA. We found that they are located in layer 2 or 3. They are the red neurons in the pictures, and most of them are located in layer 2 or 3, as shown in the panel E. And they are not GABAergic cells. They are labeled by GABAergic neurons marker GAD67. We further confirmed the direct synaptic connection using optogenetics and patch clamp recording. We injected channel dopsin virus in Bmpfc and recorded the light-evoked current in FRA. We found both D1 and D2 neurons innervate FRA neurons with excitatory and inhibitory inputs. They are identified by specific blockers. It's a little complex. And it's quantified by EPSC and iPSC amplitude. It showed to us connections are not simple. Both D1 and D2 neurons have excitatory and inhibitory projections. Then, what's the difference between D1 and D2? We try to answer it by combining in vivo calcium imaging and microinfusion of D1 or D2 receptors specific drugs. The situation is a little complicated here. And a simplified conclusion is we found both D1 and D2 receptors were involved, but they are different in the regulation of population dynamics. I didn't show the sample trace here. If you are interested, I can show you later. And we are trying to figure it out at present, why they are different. We are very interested and we want to know. So we conclude that the Bmpfc FRA projection could mediate the cocaine effect on FRA. It's also dopamine signaling dependent. And together with all the evidences, this is the final summary for this story. We found that FRA is required for locomotor sensitization, and its activity SSCS was regulated by Bmpfc FRA pathway in a dopamine-dependent way. The last is my acknowledgments to Professors Zhuanzhou, Changhe, Xiaowei, and especially to my partners Minggao and others, and what the people have in our lab. That's all. Thank you all for listening. Thank you.
Well, thank you, Dr. Wang. That was a great talk. And now let's hear it from the advisory board, please. Any questions? No questions then? May I ask a question?
Yeah, please. Thank you.
What implication do you think that your research will have on, the rehabilitations of addicts and, patients that have, similar loss of functions?
I don't need to share my screen. And the direct answer is, this is about the acute effect of cocaine. And we found the phenomena about the loss of function of locomotor sensitization. And so the significance comes from the behavior sensitization. Since the why a drug is addictive, we believe it firstly induces the plasticity change and then the circuit change. And then the animal behavior or human addicts can be addictive. And so the sensitization is believed to be related with plasticity and also underlying the later circuits change and behavior change. So it's the very beginning marker of the drug exposure. So this is the significance, yeah. Okay. But it is far away from the treatment.
We have some more questions here.
Okay, thank you.
Dr. Li Yulong asked, how is the circuit connected to the motor cortex?
Okay, first, the FRA we observed is the area near the motor cortex. So we also found from the retrograde projection, many like M2, many neurons project directly to FRA. So we also want to direct analyze. We want to know whether the motor cortex will affect the frontal association cortex. So we, in fact, observe from FRA and also motor cortex and also visual cortex. We found a universal inhibition in at least these three cortex, but we didn't find the obvious dopaminergic effect from the motor cortex to FRA cortex. So the connection exists, but the functional effect we cannot make sure. Yeah.
Dr. Zhang Yan asked, is there co-release of dopamine and glutamate of Bmpfc and VTA neurons?
Thank you. Yeah, in fact, at least in VTA neurons, the dopamine and glutamate, or dopamine and GABA, they are co-released. They are reported by several papers. However, in Bmpfc, I don't know. As far as I know, I don't know anyone reported. But in some cortical neurons, GABA, even glutamate and GABA transmitters, they can co-release from a same neurons. So in the whole brain, many kinds of things we don't know. So I'm not sure. Yeah.
One last question from Dr. Zhou Yang. What are the effects of cocaine on activity in Bmpfc?
Oh yeah, it's also a great question. We also, I plan to do it for the last half a year, but I didn't find the time to do it since we have more directly experiments like define roles of different connections between Bmpfc and FRA, since we know the connections like excitatory and GABAergic connections exist between Bmpfc and FRA. And we have just 2 days before we finished the behavior test, and we found the GABAergic projections can exceed all other kinds of projections. They dramatically influence the sensitization behavior. But we also, this is maybe the last questions for this story to make it more complete. So we just don't know how dopamine or cocaine can affect Bmpfc neurons. This needs the in vivo or free moving reporting of the mice and modulate the dopamine neurons or direct modulate the dopamine receptors positive D1, D2 positive Bmpfc neurons. We didn't do it at present. And in literature, we know some slice data. They reported that the dopamine regulation of Bmpfc neurons is very complicated. They are dependent on the dopamine concentration and also the dopamine receptor types, like whether it's D1 or D2 neurons, and also the paradoxical evidence exists. They observe the different phenomena in Bmpfc neurons, so it's relatively complex. Yeah, and we want to do it in free moving level. Yeah.
Thank you again, Dr. Wang, and thank you for all the questions. Our next speaker, the next presentation will be focusing on neurocoding of the sensory motor cortex and the interaction between sensory input and motor control. The title is neural computations through sensory motor dynamics for predictive sensory motor control. Now let's give it up for Dr. Zhang Yiheng from the Chinese Academy of Sciences Institute of Neuroscience.
Hello everyone. I'm Yiheng Zhang, a postdoctoral from Dr. Cui Lab in Institute of Neuroscience, Chinese Academic Sciences. Our lab studies the neural mechanism of motor control and the application of next generation in brain-machine interface. It's a pleasure to attend the Beijing Brain Conference and share our recent work with Yun Chen and Tianwei Wang in our lab. Today, my report is about neural dynamics for predict sensory motor control. We make suitable movements to the rich environment in our daily life. Give an example of playing ping pong. We need to observe the route of coming ball and the position of opponent, adjust the angle and the strength of beat, and choose the return route on previous experiences. Here we focus on visual guided manual interception, relevant neural control mechanism. The primary motor cortex, M1, is central part of descending pathway, and 1/3 of cortical spinal tract arises from M1. What's more, M1 have a special projection, the cortical motor neuron, which have monosynaptic connection to the motor neuron in the spinal cord. The classical study found that the M1 encode reach direction. George Paulus trained monkey to do the sent-out reach task, and record M1 neural activity. They found neural activity is related with the reach direction at cosine tuning. The direction with maximum failure rate is defined as the neuron's preferred direction, SPD. However, recent studies show that M1 is modulated by sensory information. Paulus asked monkey to do the random target pursuit task. Monkey used the robotic arm to control a cursor on the screen and to track the random position target. During the visual playback session, monkey just holding center and watch the video of moving cursor trajectory in the last active session. They found M1 neuron not only failed in the active session, but also responds in the visual playback session. And 70% of M1 neuron has similar direction tuning between two sessions. These results suggest M1 is not purely motor and has sensory motor property. And we further asked how visual motor interplay in M1. We use a flexible manual interception task. First, the monkey holds center to start the trial, and target appear in the random location and make circular motion. After delay, the center turns dark as go signal for monkey to reach for the target. Once any point around is touched, the target stopped. If hand endpoint is near the target, monkey got choose reward. There are 5 kinds of target speed, trial by trials. Clockwise and counterclockwise, high speed and low speed, and the static target. Our task could well dissociate sensory and motor information in temporal and spatial. We could say that monkey performed very well in the video, and the hand trajectory is almost directly to the target, suggesting the full prediction before reach. After behavior training, we record M1 neural activity when monkey performed the task. We use the Utah array to simultaneously record about 100 neurons, and the data were processed by offline sorting, even marker aligning and temporal smoothing as neuron failure rate. Then we use static and regression and other methods to represent the relationship between neuron activity and the task parameters. We further built some model to simulate more conditions and test some hypotheses. Here are some examples of M1 neuron with rich tuning modulated by target speed. The first neuron has PD shift with target speed. Here are rasters and the PSTH of spikes. Each subplot collects the trails with similar rich direction, rich endpoint in 45 degrees. 5 colors are 5 target speed. We could see the neuron activity is dramatically modulated by target speed, and the rich tuning around move one side have large shifts between clockwise and counterclockwise. The second neuron has gain modulated by target speed, which show in tuning curve and PSTH. In addition, moving target could change the baseline activity. The neuron baseline could have overall adjustment among conditions in the third neuron. We try to classify the population in 3 typical neurons in typical ways. However, they are highly mixed. The percentage of 3 modulation are close and with large overlap. Next, we explore the neural state with sensory motor interaction. The population data have over 700 successful trials and near 100 neurons, and the active pattern is very complex. So we use principal component analysis, PCA, a common dimensionality reduction method to reduce the neuron dimensionality in a few principal components, PCs, with great data variance, and use this easy component to describe the target feature of single trials. These PCs are neural state. We could find the neural state well represents the sensory and motor interplay. The neural states of single trials shown in the first 3 PCs. A left and right figure are same data colored in target speed condition and rich direction condition. We first show the static condition. Each dot is a single trial, and the neural state forms a circle, which could be fitted as an ellipse. We could say the ellipse of neural state is highly related with rich direction. Okay, the neural state in other conditions also form ellipses. Interestingly, they have similar rich coding in the first 2 PCs, while the slope of ellipse change with target speed in the third PC. The normal vector of ellipse are related with target speed. We also fit the PC with rich direction. The PC3 had PD shift with target speed. Here we found the neural state have clear rich direction tuning and also modulated by target speed in the third dimension. Can we use ideal neural activity to find the relationship of individual neuron to coding and the neural state? We simulate 3 kinds of interaction group. Each group have 200 neurons with PD distributed uniformly. Then we do PCA in 3 groups respectively. The simulation results show that the neural state of baseline group have clear layered. The gain and PD shift group have similar neural geometry. However, the PD shift group has PD chain in the first 2 dimensions, and other 2 groups don't have. Above all, the gain group is more like well the real data, which means the gain modulation dominates the population sensory motor interplay. Okay, next we also use the recurrent neural network RN to mimic the sensory motor interaction. We select target-related parameters as input, target location, motor intention, and the go signal, and choose the hand speed and output. We train RN to fit the input and output. The node activity of training RN has similar pattern with M1, and the neural states of RN nodes also have the similar neural geometry with the real data. If we change the formation of motor intention, the neural geometry will collapse. Here we speculate that the motor intention from PBC is important for M1 neural geometry generation. Our previous work also found PBC encodes motor intention in the interception task. The tuning curve of F7A have consistent rich direction tuning rather than target location alignment. And the tuning property of F7A has temporal change. Around target onsite, F7A is more related with target location, and around the reach onsite, F7A is more related with reach direction. This result suggests dynamic evolution of sensory motor control. Okay, we believe that M1 is part of frontal parietal loop for sensory motor computation. Our results give evidence that several brain areas simultaneously involved in the visual motor control and have dynamic interaction, rather than the classical hierarchic processes. Next, we are going to study the difference between M1 and PBC and how motor plan is formed in frontal parietal network. Okay, for summary and some take-home message, M1 single neuron are highly mixed, which represents hygienic tuning of sensory and motor. We also found neural states have ring-like structure, jointly encode the sensory and motor information. Our results suggest that M1 is profoundly involved in sensory motor computation rather than merely motor execution. Okay, that's all. Thank you for listening and welcome to questions and comments.
Thank you, Dr. Zhang. And we already have one question from the advisory board. Dr. Zhou Yang wanted to know that have you tried to fit the direction tuning for each single neuron?
Okay, yeah, of course, we did that. And let me show the slide. Oh, have you seen the slide? No. Okay. Here, yeah. Have you seen the slide? Yeah. Here's an example, 3 example neuron in our previous report, and it's a PD-shaped neuron, gain neuron, and baseline neuron. We can see that if we use the full model to describe this neuron, the full model is much better than the single fitting result. So we summary for single neuron fitting summary, we could also say that the full model could well describe the
than the PD shape gain or baseline activity. So we could here is also consistent with our single neuron tuning summary. The fitting results also consist with the highly mixed. Okay.
Okay, thank you. Any more questions? Well, then we also don't have any questions coming from the audience. So thank you again, Dr. Zhang, for your wonderful talk. We're glad to have you here.
Yeah, thank you.
All right, next up, Dr. Zhou Changping of Peking University, whose research on the role of lymphatic vessels in cardiovascular diseases and brain tumors has earned prestigious publications and even a Cell cover paper. The topic of his talk today is meningeal lymphatics regulate radiotherapy efficacy through modulating anti-tumor immunity.
Good afternoon, everyone. I'm Changping Zhou from the Institute of Molecular Medicine of Peking University. It's my honor to participate in this conference. I'm a postdoctor in Jinchai Luo's lab. Recently, Professor Luo's group highlighted a crucial role of meningeal lymphatics in anti-tumor immunity in brain tumor. Brain tumors are among the most feared of forms of cancer, but the effective therapies are lacking. Radiotherapy is one of many conventional therapies for brain tumors. Over the past decade, accumulating evidence has shown that the local irradiation of solid tumors, including gliomas, has immunoregulation effects. However, it remains unclear how radiotherapy regulates immune response and whether radiotherapy trigger anti-tumor immunity can be manipulated. For a long time, central nervous system had been commonly accepted as a system without classical or lymphatic vessels. The rediscovery of meningeal lymphatic vessels has provided a new aspect of immunity in the central nervous system. Like a bridge, meningeal lymphatic vessels make connection between the brain and the cervical lymph nodes and are open to the trafficking of immune cells such as D cells and T cells. Our previous studies and another research published in Nature clearly revealed the anti-glioma effect of immunotherapy is approached in mice with defective meningeal lymphatic vessels. Thus, we deduce that meningeal lymphatic vessel cervical lymph nodes network contributes to the efficacy of radiotherapy in glioma while modulating immune response. Furthermore, enhancing meningeal lymphatic vessel functions by Bmpfc treatment can increase the radiotherapy efficacy. To define the contribution of meningeal lymphatic vessel cervical lymph nodes network to the treatment of brain tumors by radiotherapy, we first used a cervical lymph nodes renewal model. The cervical lymph nodes collect lymph from meningeal lymphatic vessels and served as the primary genuine lymph nodes of brain tumors. We found that radiotherapy significantly prolonged the survival of mice-bearing gliomas and decreased the tumor size. In contrast, this therapeutic effect was impaired when cervical lymph nodes were removed. Besides, we found that radiotherapy could induce the anti-tumor immunity while promotion of the infiltration and activation of CD8 positive T cell in gliomas, while cervical lymph nodes removal could impair this effect. Then we evaluated the role of meningeal lymphatic vessels while meningeal lymphatic vessel defective mouse model and found similar results. Then we used another meningeal lymphatic vessel defective mouse model while for the chemical effect of withdrawal and laser exposure. And confirm above results. As expected, meningeal lymphatic vessel deficiency decreased the radiotherapy trigger anti-tumor immunity. Furthermore, we found that meningeal lymphatic vessels are the key root of increased DC trafficking in radiotherapy. Meningeal lymphatic vessel ablation may impair the radiotherapy-induced DC trafficking, which lead to the failure of T-cell priming in cervical lymph nodes. The above results demonstrate that the therapeutic effect of radiotherapy on glioma is dependent on the intact meningeal lymphatic vessels and cervical lymph nodes, which is related with anti-tumor immunity response. Given the importance of the meningeal lymphatic vessel cervical lymph node system for the radiotherapy, we hypothesize that enhanced meningeal lymphadenopathy would potentially radiotherapy-induced anti-tumor immunity. Thus, we investigate the correlation between the gene expression of lymphadenopathic effects and immune cell infiltration into the glioma of patients after radiotherapy treatment. By analyzing the database from the Chinese glioma genome atlas, results showed that Bmpfc appeared to be a unique gene that was highly correlated with CD8 positive T cell number, but had no correlation with Treg cell number. Even though Bmpfc expression did not significantly differ in glioma patients with no treatment or radiotherapy treatment alone. Collectively, these results suggest that Bmpfc levels are correlated with T-cell infiltration in glioma patients after radiotherapy. Thus, we further investigate the correlation among the Bmpfc level, radiosensitive, and immune cell infiltration in mice gliomas over expression Bmpfc or humbling vector control. We found that Bmpfc overexpression group survive longer than the vector group after radiotherapy, and the radiosensitivity of Bmpfc overexpression gliomas was oppressed after cervical lymph nodes removal. Besides, tumor-derived Bmpfc overexpression increased the CD8 positive T cell number and DC trafficking. In order to explore how Bmpfc promote DC trafficking after radiotherapy, we assessed the difference in meningeal lymphatic vessels between the Bmpfc and the vector group. As expected, the Bmpfc overexpression in tumor promote meningeal lymphatic vessel expansion. Most importantly, CCL21 expression was higher in the lymphatic and serial cells of meningeal lymphatic vessels after Bmpfc overexpression. CCL21 is the most important lymphatic and serial cell derived chemical for DC recruitment. To determine whether CCL21 pathway mediated the facilitation of anti-tumor immunity induced by the Bmpfc overexpression, IgG isotope and anti-CCL21 antibodies were each administered to mice bearing glioma that overexpressed Bmpfc. The data showed that Anti-CCL21 antibodies reduced the survival time after radiotherapy of mice bearing glomus overexpression VEGF-C. Besides, anti-CCL21 antibodies decreased the CD8 positive T cell number and DC trafficking. Collectively, CCL21 blockade reversed the increased efficacy in mice-bearing tumors overexpression VEGF-C in response to radiotherapy treatment, suggesting that VEGF-C potential and radiotherapy and this is dependent on the CCL21 pathway. VEGF-C mRNA has been shown to be a potential tool in anti-glioma immunity enhancement in mice. In this study, we designed a VEGF-C mRNA. As shown, VEGF-C mRNA induces the remodeling of miniature lymphatic vessels and promote the expression of CCL21. Most importantly, the data showed that mice treated with VEGF-C mRNA survived much longer than mice treated with control mRNA after radiotherapy. And VEGF-C mRNA promoted T cell premium in cervical lymph nodes and CD8 positive T cell infiltration in tumors. Furthermore, increased disease were detected in cervical lymph nodes from the VEGF-C mRNA group after radiotherapy. Thus, VEGF-C mRNA may be a potential therapeutic drug for radiotherapy enhancement. To expand the above findings, we also investigate the contribution of miniature lymphatic vessels cervical lymph nodes network in intracranial lung cancer metastasis model. Data showed that VEGF-C also enhanced radiotherapy trigger anti-tumor immune response in metastasized brain tumors and also enhanced the efficacy of radiotherapy. We finally analyzed the dynamics of T cell receptor CDR3 beta sequence in order to explore the change of T cell clonality after VEGF-C treatment. We compared the number of CDR3 beta clone types, which were significantly higher in VEGF-C mRNA group compared with the control mRNA group. Besides, the functional CD8 positive T cells, which produce various cytokines such as TNF-alpha, INF-gamma, and so on, were increased in tumor from VEGF-C mRNA group. This data indicate an outstanding anti-tumor immune response in VEGF-C mRNA combined with radiotherapy. Above all, we show that miniature lymphatic vessels contribute to the inhibitory effect of radiotherapy on brain tumors, mediated at least in part through the modulation of radiotherapy trigger anti-tumor immune response. The effect of radiotherapy on intracranial glomus and metastasized lung cancer was impaired after the excision of cervical lymph nodes or disruption of miniature lymphatic vessels, as evidenced by a shortened survival time and a larger tumor value in miniature lymphatic vessel disruption and cervical lymph nodes excision groups than in control mice. Taken together, these findings underscore the importance of miniature lymphatic vessel and the cervical lymph nodes network in regulation of radiotherapy modulated anti-tumor response, and suggest a working model that radiotherapy stimulated the trafficking of brain tumor-derived DC to the cervical lymph nodes and the subsequent activation of CD8 positive T cells, which is rely on intact meningeal lymphatic vessels and cervical lymph nodes network. Besides, this work has been published in Cell Research as of Carolina recently. As increasing evidence has shown uncovered the important roles of the lymphatic vessels in normal and pathological process. Thus, my next step is to further clarify the role of lymphatic in the several diseases such as brain tumors and cardiovascular disease. Thanks.
Okay, thank you, Dr. Zhou. There is a question from Dr. Chen Jian in the comment section. He said, glioma is usually considered as a cold tumor with very few T-cell infiltration, and gliomas are also very resistant to radiation therapy. Have you observed defects of MLV in glioma patients, or I guess in animal models as well? He also said, on average, a GBM tumor usually has about 100 mutations, but GL261 has more than 4,000 mutations. So it is a hot tumor model. So what's your experience on cold tumor models?
Thanks, Dr. Chen's question. Indeed, we did not observe the defective of meningeal lymphatic vessels in glioma patients. We only did it as in mice model. Thus we couldn't answer this question. But we try to make gliomas as a cold tumor into a hot tumor, because we GFCM1A could enhance the anti-tumor immunity, which could enhance the T-cell infiltration in gliomas. In question 2, indeed, there is limitations we couldn't ignore. So we use another model, LLC model, but another tumor, glioma tumor, such as delta T2T, we couldn't because we couldn't get other way only use GL261 or LLC model to perform this access. Sorry.
Right. Thank you, Dr. Chen Jian, for the question, and also thank you, Dr. Zhou, for the talk and your answers. So, is anyone tired now? It's been a very information-intense afternoon, isn't it? I hope you all perk up now, because the next talk is precisely about paying attention and how neurodivergent traits interact with the process of paying attention. Well, next up, we have someone all the way from University of Oxford. Let's welcome Dr. Gongwei Kang, and I hope it's not too early where you are, to present his findings in the multimodal computational analysis and diagnosis using MRI images and other subject information. The topic of their presentation is modeling multimodal population brain imaging data. Let's welcome Dr. Gong.
Hello, everyone. I'm very glad to have the chance to give a talk in this postdoc forum. My name is Wei Kang Gong, and I'm a postdoc researcher with Stephen Smith at the University of Oxford and Christine Beckman at the Donner Institute in Nijmegen. My work concentrates on creating tools and techniques for analyzing large-scale population brain imaging datasets. Today, my topic is modeling multimodal population brain imaging data. The neuroimaging field is facing a big data era. imaging neurosciences have seen a transformation in recent years. While initial imaging studies involves maybe 30 to 100 subjects, with projects like the Human Connectome project, the field has moved forward. Currently, as part of the UK Biobank Imaging Extension project, some 100,000 subjects are being imaged. And while such projects offers tremendous opportunities to understand the brain in terms of thinking structure, function, to clinical or cognitive or demographic variables, however, computational feasibility remains a challenge for such big datasets. Currently, the most widely used approach for compressing such imaging resources is to use image-derived phenotypes, typically scalar quantities that summarize image features into simple numbers. An example would be the mean subcortical volume derived from a subject's anatomical image, or the mean Z statistics under task within a certain cortical area derived from the functional MRI. The IDPs have been shown to have very good heritabilities in our recent paper. This knowledge-based approach is simple and efficient, and effectively reduces the high-dimensional data into interpretable, compact, and convenient features. However, there may be a large loss of information due to IDP not capturing important source of subject variabilities, as well as ignoring cross-modality relationships. And therefore, an open question remains if we can do better defining these IDPs in a data-driven fashion. Further, such large image studies deliver multiple types of data, and from these we can derive even a higher number of phenotype maps. For instance, a single diffusion experiment gave rise to multiple maps of scalar values, such as FA or mean diffusivity. Classically, these derived maps would all be analyzed separately. Our approach for simultaneously interpreting and analyzing such multiple modalities was to focus on common modes of variation across the population. Given these multimodal maps together with subjects' demographic, behavior, and clinical informations, known as non-imaging derived phenotypes, we aim to discover a low-dimensional representation of the brain maps. Each of them represents a specific set of brain areas, covariant gathers related to a given IDP. In 2010, our group introduced linked assay. In a nutshell, that method decomposes given data modalities through independent component analysis. Flickr does separate SAAs in each modality, but constrains that subject cores are shared across modalities. Therefore, for each component, there is one common subject mode with modality-specific spatial maps. In addition to this, Flickr also estimates a positive weight per component per modality, representing the contribution of a component on a given modality. Flickr achieved this by a variational Bayesian algorithm. This spatial independent constraint is achieved by a mixture model prior. For UK Biobank data in this work we analyzed so far, we have analyzed 14,000 subjects across 47 modalities, each being represented across over 150,000 voxels. Jointly, that is 800 gigabytes of raw data. If we use vanilla Flickr on this data, we would require approximately 1 gigabyte of peak memory and some 1.6 thousand hours of computation. That's larger than 2 months non-stop, even for this small subset of UK Biobank subjects. So in order to address this, we have modified Flickr and introduced two types of innovation. First, we introduce data compression in subject space. By this, I mean we analyze the subject-by-subject covariance matrix of the data and create what we can best describe as eigensubjects. This reduces the dimensionality of the matrix fed into Flickr by a controllable amount. For those interested in the detail, I'm pointing you to the relevant paper here. Secondly, we combine this with compression in the spatial domain. Instead of using fixed atlas, we use what is called dictionary learning, an approach that allows us to define a data dictionary per modality. Again, I won't have time for the technical details and only refer to a very relevant paper here. We have extensively evaluated those approaches using simulated data across different signal-to-noise levels, different level of compression, and different number of components. The big flicker with MIG-P processing gives similar results to the original flicker, and both flicker and the big flicker accurately find the underlying ground truth in most cases. On the left, the high SNR case, a relatively low dimensional MIG-P can achieve an accurate estimation of components, while on the right, the high SNR case, a high dimensional is needed. For dictionary learning, we tested the influence of signal-to-noise ratio, component numbers, dictionary dimensions, and dictionary sparsity. Figure on the bottom shows that in almost all circumstances, the optimal performance given by dictionary learning matches that of non-reduced analysis. In summary, we can obtain very significant data reduction in both in subject domain and in spatial domain through these two approaches. In real data, as there is no ground truth available, we tested whether the modes of big flicker have a similar prediction accuracy of 90Ps compared with the original flicker using 1,000 subjects from the UK biobank. Here are some blood antibody plots shows that the difference in the prediction correlation with 90Ps in the UK biobank. In almost all cases, we see a delta R that is small but larger than 0, which means that our prediction accuracy is equal or even better when compressing the data relative to finding the non-compressed data. We assume that this is due to some of the inherent denoising when compressing the data. For the computation time on the 14,000 subjects we analyzed so far, depending on the number of components to extract, vanilla flicker require up to 1,600 hours of computation and up to 1 GB of memories. Well, through our compression approach, we can fix the peak memory to 50 GB and reduce the computation time to less than 100 hours. For the full UK biobank sample, we estimated that without compression, we need some 6 GB of peak memory and 12,000 hours of computation. If we use vanilla flicker, well, with big flicker, we can obtain the decomposition in 1% of time and 1% of the memory. Ultimately, this means that instead of using 1.5 years and a supercomputer to analyze the whole dataset, we will be able to analyze 100,000 subjects within 5 days on a laptop. So in terms of the prediction, UK biobank contains over 8,000 non-imaging phenotypes. The plot shown earlier relate to analyze all of this, but I will give you a few examples. For example, big flicker has a significant improvements on prediction accuracy on fluid intelligence, on age-started wearing glasses, on handedness, and on hypertension. Further, all of these correlations can be mapped back into the brain, so we can investigate which data modality give rise to good predictions. For example, when it comes to handedness, the lateralized modes are involved, including resting state language network, task activations, surface area, and white matter tracks. Well, if you are interested in age-starting wearing glasses, we can predict this both from the task and the rest data. Several resting state connectivity and task modalities showed associations in primary visual areas, which is consistent with the fact that this is a vision-related health variable. And the approach introduced so far works in 2 stages. First, we use the optimized flicker approach to estimate subject loadings, and in the second step, we link this to NIDPs. So can we do better? And yes, we think we can. Instead of separating the decomposition and prediction stage, we can link these two via joint cost function. Not only the data reconstruction, but also the prediction parts now involves in the estimation. And the cost function now can involve more than a single NIDPs in the estimation. That is, we can use multitask learning so that we can steer the decomposition towards the features that are generically have high predictive power. Again, we have evaluated this across over 16,000 NIDPs and 14,000 subjects in the updated UK biobank subjects. Here is an example of looking into the prediction of IQ. Well, Big Flicker already does better than predicting IQs from handcraft IDPs. We observe that this is further improved by adding supervision into the cost function, which is shown in the third bar. Further, by including some NITBs that highly relate to IQ, we can further improve the prediction of the IQ by multitask learning, since the fourth and fifth columns. A similar trend is obtained for other NITBs, like hypertension, adenosis, and age-static wearing glasses. Note that these NITBs are selected because the original Big Flicker have a very good prediction performance. In summary, if Big Flicker enhance the ability of original Flicker for multimodal fusion in the large dataset, the modes learned by Big Flicker have an increased prediction accuracy of NIDPs compared with traditional methods like handcraft NIDPs, ICAs, and the original Flicker. So adding supervision to the Big Flicker further helps improve the multimodal learning. In summary, if you are interested in using our algorithm, please refer to our GitHub links. And if you are interested in more details, please refer to the 2 papers here for more details. That's all for my talk. Thank you very much for listening.
Well, thank you, Dr. Gong. Any questions from the advisory board? No questions? Well, I would like to ask a small question. Since your research was done on a British medical data corpus, do you plan on applying the same method to, for example, maybe the medical data corpus of another population of another country or something? Is this more applicable regardless of the corpus that you're working with, or is that more reliant on your neural network, and for another data corpus you have to retrain that? What do you think?
Yeah, that's a very good question. So we have tested our approach in other data sets in my current research, and they have a good performance, but haven't published. So I think the generalizability of the methods is very good. It is a general approach, I think.
That's certainly very exciting to hear.
Yeah.
Since there doesn't seem to be any more questions coming, then, well, thank you, Dr. Gong, again for coming. We're glad to have you here.
Yeah, thank you very much.
And now, if you have any additional questions for our presenters, you may connect with them through their contact information in the program. You can also connect with Cybers through our official WeChat account and Xinyi platform, as well as Bilibili channel. Now, as we are wrapping up, the awardee of today's forum is being sent to me from the advisory board, and let's give a few minutes of that. But before announcing the award, I want to take this time to remind everyone that regardless of whether you have an award or not, you all did an amazing job, and we're honored to have you here. And don't forget, a huge thank you to our advisory board members who took their precious time to be here with us. So now let's give our appreciation to Dr. Chen Jian from Chinese Institute of Brain Research in Beijing, Dr. Guo Qiang from the School of Life Sciences at Peking University, Dr. He Aibin from College of Future Technology at Peking University, Dr. Luo Minmin from Chinese Institute of Brain Research, Beijing, Dr. Li Yulong from School of Life Sciences at Peking University, Dr. Song Yan from School of Life Sciences at Peking University, Dr. Song Chen from Academy for Advanced Interdisciplinary Studies at Peking University, Dr. Wang Chu, also from Academy for Advanced Interdisciplinary Studies at Peking University, Dr. Wang Wei from School of Life Sciences at Peking University, Dr. Wang Zheng from School of Psychological and Cognitive Sciences at Peking University, Dr. Zhang Yan from School of Life Sciences at Peking University, Dr. Zhou Yang from School of Psychological and Cognitive Sciences at Peking University, Dr. Zhou Peng from College of Chemistry and Molecular Engineering at Peking University. Let's give a round of applause to our advisory board members. Thank you all for providing invaluable insights to the postdoctoral research and careers of our speakers. And I would also like to thank our staff members who worked tirelessly behind the scene, and it was a great honor to work with you today. So now let's give it a 5-minute rest before we announce the award for today. The Excellent Report Award in the 2022 Beijing Brain Conference, the Postdoctoral Forum for Advanced Interdisciplinary Studies, goes to Dr. Zhao Boxuan from Stanford University, Dr. Qian Yongjun from Duke University, Dr. Sun Huan from St. Jude's Children's Research Hospital, and Dr. Cao Yijun from Yale University. Congratulations to the awardees for their stellar presentation, and congratulations to everyone for a successful forum. Now, before we wrap up, let's give our warmest welcome to Dr. Zhou Peng, who will deliver our closing speech. Welcome, Dr. Zhou Peng.
Thank you, and thank you all. And first of all, I want to say thank you for all the speakers for your very wonderful work. I've enjoyed the talks very much today. I think each one of you is chosen out of a very large and highly competitive pool of candidates by the voting from our steering committee. I should say that due to the limited number of presentation slots available and the high quality of submitted abstracts, sometimes it really pains to make decision of choosing one versus the other. And in the end, we decided upon the 18 talks as you have seen today. From your performance, I could tell all the presentations are carefully prepared and well executed. I've learned a great deal, and I believe the audience have done. I've also learned a lot. So thank you all for coming to this online symposium. Secondly, I also want to thank the organizers and all the volunteers for their very hard work to put together this amazing symposium. As some of you may know, this is the third symposium in the series, and typically this is organized by a committee of postdoctoral fellows at Peking University with support from the Center for Life Sciences Office and an advisory board of faculty members. And this year, because this symposium is integrated into Beijing Brain Conference, we have had additional support from CIBR faculty and staff members. This is one of the most efficient and professional team that I have worked with over the past few years. I think it's really their dedication that make this happen. And last but certainly not least, I want to thank the institutes that support this symposium, including the Academy of Advanced Interdisciplinary Studies at Peking University, the Chinese Institute for Brain Research, the Center for Life Sciences, and IDG McGovern Institute. And for this symposium to really happen, this is really the result of a joint effort from PKU and CIBR. So I look forward to our continued collaboration in the future. And thank you all for staying up towards the end. I think it's time to say goodbye, and this symposium is now concluded. Thank you all very much.